It's a very constant and continuous process which happens when we are with great sages and eminent professionals. So herewith we have all organized here to witness the beam of knowledge regarding the topic, how to get an understanding regarding synopsis. So before starting with the actual session, I request Dr. Nisarga to render Dhanvantari Samana. I request all the dignitaries who are on dais to lit the lamp and grace the occasion.
ಸರ್ ಅಲ್ಲಿ ಇದ್ರ ಕೋವಿಡ್ ರಿಪೋರ್ಟ್ ಏನೋ ತಯಾರು ಮಾಡ್ತಿದ್ದಾರೆ. Yes, first. Yes. ಹಾರ ಇತ್ತಂದ ಯಾರು ಆರ್ಗನೈಸ್ ಮಾಡೋದು ಸರಿ ಮಾಡ್ಬೇಕು ಏನು ಮಾಡಿದ್ರು ಪರ್ಫೆಕ್ಟ್ ಮಾಡ್ಬೇಕು. Students ಒಂದು ಫಿಕ್ಸ್ ಒಂದು ಹೂವಿನ ಪ್ಲಾಸ್ಟಿಕ್ ಇಂದ ತಂದ ಫೋಟೋ ಕಿಟ್ಟಿಟ್ಟು
Thank you all. Now it's my honor and privilege to welcome the guest speaker of today's session, Dr. Santosh N. Belawadi, who is the son of Shri Neelappa K. Belawadi, who has done BAMS, MD, and PhD in the Department of Kaya Chikitsa. He is presently working as professor and HOD in Department of PG and PhD Studies in Kaya Chikitsa and academic dean, DGM Ayurvedic Medical College Hospital, Postgraduate Research Center, Dr. S.V. Savadi Road, Gadag, Karnataka. He has got 18 years of teaching, clinical, and research experience. He is known as a writer, clinician, teacher, researcher, doctor, as well as a poet. Languages he knows English, Hindi, Kannada, and Sanskrit. He has done his BAMS in Government Ayurvedic Medical College, Mysuru, University of Mysuru. Year of passing is 1998. MD he has done in Government Ayurveda Medical College, Mysuru, Rajiv Gandhi University of Health Sciences in the year 2003. He has done his PhD in Ayurveda Mahavidyalaya, Hubli, Karnataka in the year 2019. He has guided 20 PG research scholars. He was an examiner for undergraduates, BAMS students, Rajiv Gandhi University of Health Sciences, Bengaluru, Karnataka, examiner for MD, Rajiv Gandhi University of Health Sciences, Bengaluru. He is also a PhD examiner, LIC member, and subject expert. He worked as Project Monitoring Officer, Rajiv Gandhi University of Health Sciences, Bengaluru, Assistant Editor for Ayush Journal, Rajiv Gandhi University of Health Sciences, Editor for Ayur Anvekshana, Bilingual Quarterly Maxim, DGM AMC, Kadak. He is a Chief Investigator, worked as Principal Investigator for Project Vision Group of Sciences and Technology, Biotechnology State Government Project to strengthen the infrastructure of the Research Institute. Recognized PhD guide, presently member BOS Postgraduate Rajiv Gandhi University of Health Sciences, Bengaluru, Karnataka. His personal significant achievements, states national awards are as follows. Best Academic Achievement Award by DGM Ayurvedic Medical College Hospital, Gadag, felicitated in state level seminar on Khagendra 2015 for authorizing book on Prativisha and Vishachikitsa in Ayurveda by Bapuji Ayurvedic Medical College Hospital, Chelkere. He is a Best PG Scholar of GIMC Mysuru during 2003. Best Paper Award Ayur Vision 2012 at Kasturba Ayurveda Medical College, Manipal. Awarded Best Paper Presentation Vaishwanara 2015 National Level Conference on Grahani Dosha, JSS Ayurveda Medical College, Mysuru. He has also got Shrestha Vaidya Sahiti Award by Health Minister Dr. K. Sudhakar and Chancellor Dr. S. Sachidanand on 5th November 2020 at Rajiv Gandhi University of Health Sciences, Bengaluru, Karnataka. Chief Person for Kannada Vaidyakiya Sahitya Sammelana at RGHS, Bengaluru. Awarded for academic contribution at Ayurveda Mahavidyalaya, Hubli, February 2022. He is a resource person for many state, national, international conferences and workshops. He has done various surveys and health checkup camps in flood-affected places near Gadag district, many health checkup camps in interior backward villages. He has published many international, national, and state peer-reviewed journals. To propagate Ayurveda, to create Ayurveda awareness, health awareness articles has been published in local Kannada regional language newspapers and state-level health magazines. He participated as a resource person in international, national, state-level conferences. He participated in interactive workshops, deliberated different health talks to common public in society in diabetes, obesity, and so on. Yes, yes. Yes, yes. Last to say, his area of interest is in Twakvikara, Vatavyadhi, and Rasayana. I am very much honored, delighted, and privileged to welcome Dr. Santosh Belawadi, sir, on this August gathering. A big round of applause. I request Dr. Rajshekar Ganiga to say a few words about Dr. Santosh Belawadi, sir.
अखंड मंडलाकारं व्याप्तमेन चराचरं तत्पदं विदितेन तस्मै श्री गुरवे नमः. ಆಯುರ್ವೇದದ ಗುರು ಪರಂಪರೆಗೆ ನನ್ನ ಸಾಷ್ಟಾಂಗ ನಮಸ್ಕಾರಗಳನ್ನ ತಿಳಿಸ್ತಾ ಈ ಒಂದು ಕಾರ್ಯಕ್ರಮದ ಅಧ್ಯಕ್ಷತೆಯನ್ನು ಸ್ವಲ್ಪ ಮಹಾವಿದ್ಯಾಲಯದ ಇವತ್ತಿನ ಪ್ರಭಾರಿ ಪ್ರಾಚಾರ್ಯರಾದ ಡಾಕ್ಟರ್ ಶಾಂತಿಕಿಶೋರ್ ಸಿಂಗ್ ಅವರೇ, ಮಹಾವಿದ್ಯಾಲಯದ ಸ್ನಾತಕೋತ್ತರ ಕಾಯ್ದೆ ವಿಭಾಗದ ಮುಖ್ಯಸ್ಥರು ಮತ್ತು ನನ್ನ ಸ್ನೇಹಿತರಾದಂತಹ ಡಾಕ್ಟರ್ ಮಾಧವಿಕಾ, ಇಷ್ಟೊತ್ತು ಅವರ ಪರಿಚಯವನ್ನೇ ನೀಡಿದ್ರು. ಅದಕ್ಕಿಂತ ಹೆಚ್ಚಾಗಿ ಮಿಗಿಲಾಗಿ ಅವರೊಬ್ಬ ಶಿಕ್ಷಣ ತಜ್ಞ, ಒಬ್ಬ ಶ್ರೇಷ್ಠ ಸ್ನೇಹಜೀವಿ, ಜೊತೆಗೆ ನಮ್ಮ ಸರ್ಕಾರಿ ಆಯುರ್ವೇದ ಮಹಾವಿದ್ಯಾಲಯದ ಅಲುಮಿನಿ ಅನ್ನುವಂಥದ್ದು ನಾನು ಬಹಳಷ್ಟು ಹೆಮ್ಮೆಯನ್ನು ಹೇಳ್ಕೊಳ್ಬೇಕು. ನಮ್ಮ 4 ಸರ್ಕಾರಿ ಕಾಲೇಜುಗಳು ನಮ್ಮ ಕಾಲೇಜುಗಳೇ, ಅದು ಯಾವುದೋ ಒಂದೊಂದಿಲ್ಲ. ಬಳ್ಳಾರಿ ಬೇರೆ, ಮೈಸೂರು ಬೇರೆ, ಬೆಂಗಳೂರು ಬೇರೆ, ಶಿವಮೊಗ್ಗ ಬೇರೆ, ಕಾಲೇಜು. ಇವತ್ತು ನಾಳೆ ನಾಳೆ ಹೋಗ್ಬೋದು, ಅಲ್ಲಿವರೆಗೂ ಬರ್ಬೋದು. ಸ್ನಾತಕೋತ್ತರ ಮತ್ತು ತರಗತಿಯನ್ನು ಎರಡೂ ಕೂಡ ನಮ್ಮ ಸರ್ಕಾರಿ ಆಯುರ್ವೇದ ಮಹಾವಿದ್ಯಾಲಯದಲ್ಲಿ ಓದಿರುವಂತವ್ರು ಸಂತೋಷ ಪಡಬೇಡಿ. ಅದಕ್ಕೆ ನನಗೆ ಬಹಳಷ್ಟು ಸಂತೋಷ ಇದೆ ಮತ್ತು ಹೆಮ್ಮೆ ಕೂಡ ಇದೆ. ಜೊತೆಗೆ ಇನ್ನೊಂದು ಹೆಮ್ಮೆ ಏನಂದ್ರೆ ಅವರು ಗದಗಿನವರು. ಅದರಲ್ಲೂ ವಿಶೇಷವಾಗಿ ನಾನು ಓದಿರ್ತಕ್ಕಂಥ ನನ್ನ ಊರು ಕೂಡ ಗದಗ ಮತ್ತು ನಾನು ಓದಿರುವಂತಹ ಏನು ಡಿಜಿ ಮೇಲ್ಮಳಿಗೆ ಆಯುರ್ವೇದ ಮಹಾವಿದ್ಯಾಲಯದಲ್ಲಿ ಅವರು ಅಧ್ಯಾಪಕರ ಕೆಲಸ ಮಾಡ್ತಾ ಇದ್ದಾರೆ. ನನ್ನ ಹಾಗೆ ಸಾಕಷ್ಟು ವಿದ್ಯಾರ್ಥಿಗಳನ್ನ ಹೇಗೆ ಅಲ್ಲಿಂದ ಕಲ್ತ್ಕೊಂಡಿದ್ದಾರೆ, ಅವರು ಮಾರ್ಗದರ್ಶನ ಮಾಡ್ತಿದ್ದಾರೆ. ಅದು ಕೂಡ ಇನ್ನೊಂದು ಹೆಮ್ಮೆ ಅನ್ನುವಂಥದ್ದನ್ನ ನಾನು ಈ ಸಂದರ್ಭದಲ್ಲಿ ಹೇಳೋದಕ್ಕೆ ಇಷ್ಟಪಡ್ತೀನಿ. ನಾನು ವಿದ್ಯಾರ್ಥಿ ದಶೆಯಿಂದನೂ ಕೂಡ ಸಂತೋಷ್ ಬೆಳವಡಿ ಅವ್ರಿಗೆ ಅಷ್ಟೊಂದು ಚಿರಪರಿಚಿತದಲ್ಲಿ ಅಂದ್ರೂ ಕೂಡ ಅವ್ರ ಬಗ್ಗೆ ನನ್ಗೆ ಚೆನ್ನಾಗಿ ಗೊತ್ತು. ನನಗೆ ಇವತ್ತು ನೆನಪಿದೆ, ನಾವು ಅವ್ರ ಶಾಪ್ಗಳಿದೆ ಇದರಲ್ಲಿ, ಇವರು ತಂದೆಯವರಾಯ್ತು, ಇವ್ರ ಅಣ್ಣ ಅವರೆಲ್ಲ ಒಳ್ಳೆ ಸ್ನೇಹಿತರು. ನಮ್ಮ ಈ BN ಮಾರ್ಕೆಟ್ ಅಲ್ಲಿ ಹೋಗ್ಬೇಕಾದ್ರೆ ಅವ್ರ ಶಾಪು ಅಲ್ಲಿ ಹೋದ ಸಂದರ್ಭದಲ್ಲಿ ನಾವು BMS ವಿದ್ಯಾರ್ಥಿಗಳು ಅಂತ ಹೇಳ್ಕೊಂಡು ನನಗಿಂತ 2-3 ವರ್ಷ ಜೂನಿಯರ್ ಅಷ್ಟೇ ಹೋದಾಗ ಅವರು ಅತ್ಯಂತ ಹೆಮ್ಮೆಯಿಂದ ಅವ್ರ ತಂದೆಯವರು ಅವರನ್ನ ಹೇಳೋರು, ಸರ್ ನಮ್ಮ ಬ್ರದರ್ ಕೂಡ ಮೈಸೂರು ಗೌರ್ಮೆಂಟ್ ಕಾಲೇಜಲ್ಲಿ BMS ಮಾಡ್ತಾ ಇದ್ರಂತೆ ಸಂದರ್ಭದಲ್ಲಿ ನಾನು ನೋಡಿರ್ಲಿಲ್ಲ. ನಂತರ ನಮ್ಮ ಒಂದು ವ್ಯವಸ್ಥೆಯಲ್ಲಿ ಪರಿಚಯ ಆಯ್ತು ಮತ್ತು ಇಷ್ಟೊಂದು ಅಗಾಧ ಪ್ರಮಾಣದಲ್ಲಿ ನಮ್ಮ ಸಂತೋಷ್ ಬೆಳವಡಿ ಬೆಳಿತಾರಂತ ನಾನು ಯಾವತ್ತೂ ಕೂಡ ಅಂದ್ಕೊಂಡಿರ್ಲಿಲ್ಲ. ಇದು ವಾಸ್ತವ ಕೂಡ. ಯಾಕಂದ್ರೆ ಅಷ್ಟೊಂದು ಜನರೊಟ್ಟಿಗೆ ಆಗ್ಲಿ ಅಥವಾ ಔಟ್ಸ್ಪೋಕನ್ ಮುಖಾಂತರ ವ್ಯಕ್ತಿ ಅಲ್ಲ, ಅತ್ಯಂತ ಮೃದು ಸ್ವಭಾವಿ, ಮಿತ ಮಾತುಗಾರ. ಅಂಥದ್ದು ಒಂದು ಅದ್ಭುತವಾಗಿ ಒಂದು ಆಯುರ್ವೇದ ಕ್ಷೇತ್ರಕ್ಕೆ, ಅದರಲ್ಲೂ ವಿಶೇಷವಾಗಿ ಆಯುರ್ವೇದ ಸಾಹಿತ್ಯ ಕ್ಷೇತ್ರದಲ್ಲಿ ಸಾಕಷ್ಟು ಜನ ಜ್ಞಾನವನ್ನು ಹೊಂದಿರತಕ್ಕಂತ ಮೇಧಾವಿಗಳಿದ್ದಾರೆ. ಆದ್ರೆ ಪುಸ್ತಕ ಬರೆಯುವಂಥದ್ದು ಬಹಳ ಕಡಿಮೆ. ನಮ್ಮ ಮಾದದಿಗ ಇದು ಕಡಿಮೆ ಅದ್ರಿ, ಯಾವ್ದಕ್ಕೂ ಕಡಿಮೆ ಇಲ್ಲ. ಆದ್ರೆ ಆ ಕುಂತು ಬರೆಯೋದಿದೆಯಲ್ಲ, ಕುಂತು ಬರೆಯುವಂತದ್ದು ಇದೆಯಲ್ಲ, ಅದು ಎಷ್ಟೋ ಜನಕ್ಕೆ ಆಗೋದಿಲ್ಲ. ನಾವು ವಸ್ತ್ರದವ್ರಿಗೆ ಹೇಳ್ತಾ ಇದ್ದೆ, ಮಾದದಿಗ ಅವಾಗ ಹೇಳ್ತಿದ್ದೆ, ಇವತ್ತು ಹೇಳ್ತಾ ಇದ್ದೇನೆ. ಅವ್ರು ಬರ್ದಿದ್ರೆ ಇಷ್ಟಕ್ಕೆ ಒಂದು 3 ರೂಪಾಯಿ ಬರ್ತಾ ಇದ್ದೇನೆ. ಅದಕ್ಕೆ ಆ ಪರಿಶ್ರಮ, ಆ ಡೆಡಿಕೇಶನ್, ಆ ಶ್ರದ್ಧೆ ಮತ್ತು ಅದಕ್ಕೆ ಸಮರ್ಪಣಾ ಭಾವ ಇದೆಯಲ್ಲ, ಅದು ಎಲ್ರಿಗೂ ಕೂಡ ಬರುವಂತದ್ದಲ್ಲ. ಅದು ಸುಮಾರು ಎಷ್ಟೋ ಪುಸ್ತಕಗಳನ್ನು ಇವತ್ತು ಹೊರಗೆ ತಂದಿದ್ದಾರೆ. ಇವತ್ತು ರಾಷ್ಟ್ರೀಯ ಅಂತಾರಾಷ್ಟ್ರೀಯ ಮಾರುಕಟ್ಟೆಯಲ್ಲಿ ಅವಕ್ಕೆ ಮುಗಿಬಿದ್ದು ಅದನ್ನ ಖರೀದಿ ಮಾಡಿ ಓದ್ತಕ್ಕಂತ ಒಂದು ವ್ಯವಸ್ಥೆ ಏನಾದ್ರು ಇದೆ ಅಂದ್ರೆ ಸಂತೋಷ್ ಬೆಳವಡಿ ಅವರ ಒಂದು ಪರಿಶ್ರಮ ಕಾರಣ ಅಂತ ಹೇಳಿದ್ರು ಕೂಡ ತಪ್ಪಾಗಲಿಕ್ಕಿಲ್ಲ. ನಾನು ಫೇಸ್ಬುಕ್ ಅಲ್ಲಿ ಸಾಮಾನ್ಯ ನೋಡ್ತಾ ಇರ್ತೀನಿ, ಒಂದು ತಿಂಗಳಲ್ಲಿ 10ರಿಂದ 15 ಪೋಸ್ಟ್ಗಳು ಅವರು ಈ ರೀತಿಯಾಗಿ ಬೇರೆ ಬೇರೆ ಕಾಲೇಜ್ಗಳಿಗೆ ಹೋಗಿ ಆಯುರ್ವೇದದ ಜ್ಞಾನವನ್ನ ವಿದ್ಯಾರ್ಥಿಗಳಿಗೆ ಕೊಡ್ತಕ್ಕಂಥ ಕೆಲಸ ಮಾಡ್ತಾ ಇದ್ದಾರೆ. ಅದು ಅಷ್ಟೊಂದು ಸುಲಭವಾಗಿರುವಂತ ಕೆಲಸ ಅಲ್ಲ. ನಂಗೆ ನೋಡಿ, ನಾನು ಒಂದು ವಾರದಲ್ಲಿ ಬಿಸಿ ಇದ್ದೆ. ಒಂದ್ ದಿವಸ ಬೆಂಗಳೂರು ಹೋಗಿ ಬಂದಿದ್ದೆ, ನಂಗೆ ಸಾಕಾಗ್ಬಿಟ್ಟಿದೆ. ಇವತ್ತು ಕಾಲೇಜಿಗೆ ಬರೋದು ಬೇಡ ಅಂತ ಕೂತಿದ್ದೆ. ಅದನ್ನ KMRC ಪ್ರಾಜೆಕ್ಟ್ ಕೊಡ್ಬೇಕಂತ DC ಅವ್ರ ಕಡೆಯಿಂದ ಫೋನ್ ಬಂದಾಗ್ತಾ ನಾನು ಮತ್ತೆ ಕಾಲೇಜಿಗೆ ಬರ್ಬೇಕಾಗ್ತದೆ. ಅಂತ ಒಂದು ಪರಿಸ್ಥಿತಿಯಲ್ಲಿ ಒಂದ್ ದಿವಸ ಮಂಗಳೂರಲ್ಲಿ ಇರ್ತಾರೆ, ಇನ್ನೊಂದ್ ದಿವಸ ಮಂಗಳೂರಲ್ಲಿ ಇರ್ತಾರೆ, ಬೆಂಗಳೂರಲ್ಲಿ ಇರ್ತಾರೆ, ಆಮೇಲೆ ದಿಲ್ಲಿ ಅಂತೆ, ಜಾಮ್ನಗರ್ ಅಂತೆ, ಜೈಪುರ್ ಅಂತೆ, ಈಗ ಓಡಾಡ್ಕೊಂಡು ಆಯುರ್ವೇದದ ಕ್ಷೇತ್ರದ ಒಂದು ಪಸರುವಿಕೆಯನ್ನ ಅಥವಾ ಒಂದು ವಿಸ್ತಾರವನ್ನ ಮಾಡುವಂತದ್ದು ಇದೆಯಲ್ಲ, ಅದಕ್ಕೆ ಸಂತೋಷ್ ಬೆಳವಡಿ ಅಂತ ಒಂದು ಶ್ರೇಷ್ಠವಾಗಿರ್ತಕ್ಕಂಥ ಕಮಿಟ್ಮೆಂಟ್ ಇರುವಂತಹ ವ್ಯಕ್ತಿಗಳಿಂದ ಮಾತ್ರ ಸಾಧ್ಯ ಅನ್ನುವಂತ ಮಾತನ್ನ ಈ ಸಂದರ್ಭದಲ್ಲಿ ಹೇಳೋದಕ್ಕೆ ಇಷ್ಟಪಡ್ತೇನೆ. ಜೊತೆಗೆ ನಮ್ಮ ಮಹಾವಿದ್ಯಾಲಯ ಕೂಡ ಈಗ 25 ವರ್ಷಗಳಿಂದ ನಾವು ನಮ್ಮ ರಸಶಾಸ್ತ್ರ ವಿಭಾಗದಲ್ಲಿ ನಾವು ಸ್ನಾತಕೋತ್ತರ ಕೇಂದ್ರವನ್ನ ಹೊಂದಿದ್ದೀವಿ ಮತ್ತು ಸಾಕಷ್ಟು ಶ್ರೇಷ್ಠ ವಿದ್ವಾನರನ್ನ ಈ ಮಹಾವಿದ್ಯಾಲಯದಿಂದ ಈ ಸಮಾಜಕ್ಕೆ, ಈ ದೇಶಕ್ಕೆ ಕೊಡ್ತಕ್ಕಂಥ ಕೆಲಸವನ್ನ ನಾವು ಕೂಡ ಮಾಡಿದೀವಿ. ಅದರಲ್ಲಿ ಈಗ ನನ್ನ ಎದುರಿಗೆ ಕೂತಿರ್ತಕ್ಕಂಥ ಒಬ್ಬ ನಮ್ಮ ಅಬ್ದುಲ್ ಕರೀಂ ಅಂತ ನನ್ನ ವಿದ್ಯಾರ್ಥಿ, ನಮ್ಮ UG PG ಇಲ್ಲೇ ಮಾಡಿ PhD ಮಾಡ್ಕೊಂಡು ಒಳ್ಳೆ ಸ್ಥಾನದಲ್ಲಿ ಅಧ್ಯಾಪಕರು ತೊಡಗಿಸ್ಕೊಂಡಿರ್ತಕ್ಕಂಥ ಸಾವಿರಾರು ವಿದ್ಯಾರ್ಥಿಗಳನ್ನ ಈ ನಾಡಿಗೆ ಕೊಟ್ಟಂತ ಕೀರ್ತಿ ನಮ್ಮ ಮಹಾವಿದ್ಯಾಲಯಕ್ಕೆ, ನಮ್ಮ ರಸಶಾಸ್ತ್ರ ವಿಭಾಗಕ್ಕೆ ಸಿಗ್ತಾರೆ. ಅಂತದ್ರಲ್ಲಿ ನಾವು ಕಳೆದ 4 ವರ್ಷದ ಹಿಂದೆ ಮತ್ತೆ 3 ಸ್ನಾತಕೋತ್ತರ ತರಗತಿಗಳನ್ನ ಪ್ರಾರಂಭ ಮಾಡುವಂತ ಒಂದು ಪ್ರಯತ್ನವನ್ನ ಮಾಡಿದ್ವಿ. ಅದರಲ್ಲಿ ಕಾಯ್ಚಿಕಿತ್ಸಾ, ಪಂಚಕರ್ಮ, ಶಲ್ಯ ಕೇವಲ 3 ವರ್ಷದಲ್ಲಿ ಒಂದು ಮೇರುಗತಿಯಲ್ಲಿ ಬೆಳೀತಾ ಇರತಕ್ಕಂತ ಒಂದು ಉತ್ತಮವಾಗಿರ್ತಕ್ಕಂಥ ಸಂಸ್ಥೆ ಅನ್ನುವಂತ ಹೆಸರನ್ನ ನಾವು ತೆಗೆದುಕೊಳ್ಳೋದಕ್ಕೆ ಸಾಧ್ಯ ಆಗಿರುವಂತದ್ದು ಡಾಕ್ಟರ್ ಮಾಧವಿಕವೇ ಆಗಿರ್ಬೋದು, ನಮ್ಮ ಶಲ್ಯ ವಿಭಾಗ ಶಿವರ್ಕೇರಿ ಆಗಿರ್ಬೋದು, ಕೆಂಡಮಠ ಆಗಿರ್ಬೋದು, ಸಂಪತ್ ಕುಮಾರ್ ಆಗಿರ್ಬೋದು, ಇಲ್ಲಿ ಕೂತಂತ ನಾ ಹೆಸರು ತಗೊಂಡಿಲ್ಲ ಅಂತಲ್ಲ, ಇಲ್ಲಿ ಕೂತಿರ್ತಕ್ಕಂಥ ಎಲ್ಲ ನಮ್ಮ ರಾಜೇಶ್ ಆಗಿರ್ಬೋದು, ಎಲ್ಲರ ಒಂದು ಪರಿಶ್ರಮದಿಂದ 3 ವರ್ಷದಲ್ಲಿ ಒಂದು ಉತ್ತಮವಾಗಿರ್ತಕ್ಕಂಥ ಒಂದು ಸ್ನಾತಕೋತ್ತರ ಕೇಂದ್ರ ಅನ್ನುವಂತ ಒಂದು ಪ್ರಯತ್ನ ಮಾಡುವಲ್ಲಿ ನಾವು ನಮ್ಮ ಒಂದು ಪ್ರಯತ್ನವನ್ನ ಮಾಡಿದೀವಿ. ಅದರಲ್ಲಿ ಸಾಕಷ್ಟು ರ್ಯಾಂಕ್ಗಳು ಬಂದಿದ್ದಾವೆ. ನಮ್ಮ ಆಸ್ಪತ್ರೆಯ ರೋಗಿಗಳ ಸಂಖ್ಯೆ ಹೆಚ್ಚಾಗಿದೆ. ಮತ್ತು ಸಮಾಜದಲ್ಲಿ ನಮ್ಮ ರೆಕಗ್ನೈಷನ್, ನಮ್ಮನ್ನು ಗುರುತಿಸುವಿಕೆ ಜಾಸ್ತಿ ಆಗಿದೆ. ಆ ಗುರುತಿಸುವಿಕೆಯ ಕಾರಣದಿಂದಲೇ ಇವತ್ತು ಇಷ್ಟೊಂದು ದೊಡ್ಡ ಇನ್ಫ್ರಾಸ್ಟ್ರಕ್ಚರ್ ನಾವು ಮೂಲಭೂತ ಸೌಕರ್ಯವನ್ನು ನಿಮ್ ಮುಂದೆ ಇಡೋದಕ್ಕೆ ಸಾಧ್ಯ ಆಗಿದೆ. ಮತ್ತೆ 29 ಕೋಟಿ, ಬಳ್ಳಾರಿಗೆ 10 ಕೋಟಿ, ಸೆಂಡೂರಲ್ಲಿ ನಾವು ಪೆರಿಫೆರಲ್ ಸೆಂಟರ್ ಅನ್ನು ಮಾಡ್ತಾ ಇದೀವಿ. ಇನ್ನು 2 ವರ್ಷದಲ್ಲಿ 10 ಕೋಟಿ ರೂಪಾಯಿಗಳನ್ನ ವಿನಿಯೋಗಿಸಿ ಒಂದು ಪೆರಿಫೆರಲ್ ಸೆಂಟರ್ ಅನ್ನು ಪ್ರಾರಂಭ ಮಾಡಿ ಅಲ್ಲಿ 6 ಜನ ಡಾಕ್ಟರ್ಗಳು, ಸುಮಾರು ಒಂದು 20-25 ಜನ ಸಿಬ್ಬಂದಿಯನ್ನ ನೇಮಕ ಮಾಡಿ ನಮ್ಮ ಮಹಾವಿದ್ಯಾಲಯದಿಂದ ನಡೆಸಿ ಗಡಿ ಬಾಧಿತ ಪ್ರದೇಶಗಳಲ್ಲಿ ಆಯುರ್ವೇದದ ಕಂಪನ್ನು ಹಡಿಸ್ತಕ್ಕಂಥ ಒಂದು ಪ್ರಯತ್ನವನ್ನು ನಾವು ಮಾಡುವಂತರಿದ್ದೀವಿ. ಅದಕ್ಕೆ ನಾವೆಲ್ಲರೂ ಕೂಡ ತಯಾರಾಗಬೇಕಾಗ್ತದೆ ಮುಂದಿನ ದಿವಸಗಳಲ್ಲಿ. ಮತ್ತೆ ಈ ಮಹಾವಿದ್ಯಾಲಯಕ್ಕೆ 29 ಕೋಟಿ ಮಂಜೂರಾಗಿರುವಂತದ್ದು ನಾನು 2-3 ಸಂದರ್ಭದಲ್ಲಿ ಹೇಳಿದ್ದೀನಿ. ಇವತ್ತು ಅದ ಪ್ರಪೋಸಲ್ ಕೂಡ ಕೊಟ್ಟು ಹೇಳಿದ್ದಾರೆ. ಇವತ್ತು ಪ್ರಪೋಸಲ್ ಇವತ್ತು ಸಲ್ಲಿಕೆ ಆದ್ರೆ ಇನ್ನೊಂದು 4-5 ತಿಂಗಳ ಕಾಲದ ಅವಧಿಯಲ್ಲಿ ಅದು ಅಡ್ಮಿನಿಸ್ಟ್ರೇಟಿವ್ ಅಪ್ರೂವಲ್ ಆಗ್ಬಿಟ್ರೆ ಇದ್ರ ಮೇಲೆ 2 ಫ್ಲೋರ್, ಆ ಬಿಲ್ಡಿಂಗ್ ಮೇಲೆ 2 ಫ್ಲೋರ್, ಹಿಂದೆ ರಸಶಾಸ್ತ್ರ ಭವನ, ಮತ್ತೆ ಮುಂದೆ ಓಲ್ಡ್ ಬಿಲ್ಡಿಂಗ್ ಇದೆಲ್ಲ ಅದು ಡೆಮಾಲಿಷನ್ ಮಾಡುವಂತ ಒಂದು ವ್ಯವಸ್ಥೆ ಆಗ್ತಿದೆ. ಹಾಗೆ ಅಲ್ಲಿ G+5 ಬಿಲ್ಡಿಂಗ್ ಅನ್ನ ಕಟ್ಟಿ ಸಂಪೂರ್ಣವಾಗಿ ಯುಜಿ ಸೆಟಪ್ ಇಲ್ಲಿ ಹಾಸ್ಪಿಟಲ್ ಅಲ್ಲಿ ಮಾಡುವಂತ ಒಂದು ವ್ಯವಸ್ಥೆಯನ್ನು ಮಾಡಿ, ಜೊತೆಗೆ ವಿದ್ಯಾರ್ಥಿನಿಯರಿಗೆ ಒಂದು 5 ಕೋಟಿ ರೂಪಾಯಿ ವೆಚ್ಚದಲ್ಲಿ ಒಂದು ಹಾಸ್ಟೆಲ್ ನ ಕಟ್ಟುವಂತ ಒಂದು ವ್ಯವಸ್ಥೆಯನ್ನು ನಾವು ಮಾಡ್ತಾ ಇದೀವಿ. ಅದರಲ್ಲಿ ಒಂದು ತಳಹಂತದ ಮೊದಲನೇ ಮಹಡಿ ಇರ್ತದೆ. ಅದರಲ್ಲಿ 2 ಬೆಡ್ ಮತ್ತು ಅಟ್ಯಾಚ್ ಬಾತ್ರೂಮ್ ಇರ್ತಕ್ಕಂತ ಸ್ನಾತಕೋತ್ತರ ವಿದ್ಯಾರ್ಥಿನಿಯರಿಗೋಸ್ಕರನೇ ಒಂದು ಹಾಸ್ಟೆಲ್ ಮಾಡುವಂತ ಒಂದು ಪ್ರಯತ್ನವನ್ನ ನಾವು ಮಾಡ್ತಾ ಇದೀವಿ. ಅದು ಬಂದು ಇರೋದು ಬಿಡೋದು ನೀವು ಬಿಟ್ಟಿರೋದು. ಚಾರನಾಥ್ ಕಾಲೇಜಲ್ಲಿ ಈ ವ್ಯವಸ್ಥೆ ಇಲ್ಲ ಅನ್ನುವಂತ ಪ್ರಶ್ನೆನೇ ಬರಬಾರ್ದು. ಅದಕ್ಕೆ ಅಟ್ಯಾಚ್ ಬಾತ್ರೂಮ್ ಇರತಕ್ಕಂಥದ್ದು 2 ಕಾಟ್ಗಳು ಇರುವಂತದ್ದು, ಅಟ್ಯಾಚ್ ಬಾತ್ರೂಮ್ ಇರುವಂತದ್ದು ಒಂದು 15 ರೂಮ್ಗಳನ್ನ ನಾನು ಕನಿಷ್ಠ ಮಾಡ್ತಕ್ಕಂಥ ಒಂದು ವ್ಯವಸ್ಥೆ ಮಾಡೋದಕ್ಕೆ ಎಲ್ಲ PWD ಸೂಚನೆ ಕೊಟ್ಟಿದ್ದೀವಿ. 2 ಫ್ಲೋರ್ ಮೇಲೆ ಏನ್ ಬರ್ತದೆ ಯುಜಿಯವರಿಗೆ, ಕೆಳಗಿನ 1 ಫ್ಲೋರ್ ಪಿಜಿಯವರಿಗೆ ಕೊಡುವಂತ ವ್ಯವಸ್ಥೆನ ಮಾಡುವಂತ ಅದೊಂದು ಕೊರತೆ ನನಗೆ ಕಾಣ್ತಿದೆ. ನನಗೆ ಎಲ್ಲರೂ ಹೇಳ್ತಾರೆ ಸಾಕೋತ್ರ ಹುಡುಗಿಯರಿಗೆ ಅಥವಾ ಇವ್ರಿಗೆ ಇದಿಲ್ವಾ ಅಂತ. ಸೊ ಅದಕ್ಕೆ ನೀವು ಎಷ್ಟು ಇರ್ತಾರೆ ಗೊತ್ತಿಲ್ಲ, ನಮ್ಗೆ ಇರೋದೇ ಡೌಟ್ ಇದೆ ನನಗೆ. ಯಾಕಂದ್ರೆ ನೀವು ಏನಂತಾರೆ ಅದು ಒಂದು ರಿಸ್ಟ್ರಿಕ್ಷನ್ಸ್ ಆಗ್ಬಿಡುತ್ತೆ ಅಲ್ಲಿ ಅಂತ ಸಂದರ್ಭದಲ್ಲಿ ನಿಮ್ಗೆ ಎಷ್ಟೊಂದು ಸ್ವಾತಂತ್ರ್ಯಕ್ಕೆ ಧಕ್ಕೆ ಬರ್ತದೋ ಗೊತ್ತಿಲ್ಲ. ಆದ್ರಿಂದ ಕಟ್ಟುವಂತ ವ್ಯವಸ್ಥೆ ನಾವು ಮಾಡ್ತೀವಿ, ಇರುವಂತ ಬಿಟ್ಟಿರೋದು ನೀವು ಇಲ್ಲ ಅಂದ್ರೆ ಯೂಶಲಿ ಕೊಡ್ತೀವಿ ಅಷ್ಟೇ. ಸೊ ಹೀಗಾಗಿ ಈ ಎಲ್ಲಾ ಕೆಲಸಗಳು ನಡೀತಾ ಇದೆ. ಇಂಥ ಸಂದರ್ಭದಲ್ಲಿ ಶೈಕ್ಷಣಿಕ ಚಟುವಟಿಕೆಗಳಿಗೆ ಒತ್ತು ಕೊಡಿ ಅನ್ನುವಂಥದ್ದನ್ನ ಪದೇ ಮಾನ್ಯ ಪ್ರಾಚಾರ್ಯರು ಮತ್ತು ನಾನು ಸಾಕಷ್ಟು ಸಂದರ್ಭದಲ್ಲಿ ನಮ್ಮ ಅಧ್ಯಾಪಕರು ಹೇಳ್ತಾ ಇರ್ತೀವಿ. ಯಾಕಂದ್ರೆ ಮಾಧವಿ ಕವಿ ಮತ್ತು ಕೆಲವೊಬ್ಬರಿಗೆ ಮಾತ್ರ ಸ್ನಾತಕೋತ್ತರ ಒಂದು ಇದ್ರದ್ದು ಅನುಭವ ಇರೋದ್ರಿಂದ ನನ್ನ ಆದಿಯಾಗಿ ಇಲ್ಲಿ ನಾನೇನು ಸೀನಿಯರ್ ಮೋಸ್ಟ್ ಅಂತ ಇದ್ರು ಕೂಡ ನನ್ನ ಆದಿಯಾಗಿ ನಮ್ಗೆ ಸ್ನಾತಕೋತ್ತರ ಒಂದು ತರಗತಿಗಳಲ್ಲಿ ಅಥವಾ ಸ್ನಾತಕೋತ್ತರ ವಿಷಯಗಳಲ್ಲಿ ಅನುಭವದ ಕೊರತೆ ಇರೋದ್ರಿಂದ ಸಾಕಷ್ಟು ಸಂದರ್ಭದಲ್ಲಿ ಸ್ವಲ್ಪ ನಾವು ಇದರಲ್ಲಿ ಆಮೇಗತೆಯಿಂದ ನಡೀತಾ ಇರ್ಬೋದು. ಆಮೇಗತೆಯಿಂದ ನಡೀತಿದೀವಿ, ಮಲ್ಕೊಂಡಿಲ್ಲ ನಾವ್ ಯಾರು ಕೂಡ. ನಮ್ಮ ತರದ ಪದ್ಧತಿ ಇಲ್ಲಂದ್ರೂ ಕೂಡ ಬೇರೆಯವರಿಂದ ಕರೆಸಿ ಹೇಳುವಂತ ಪ್ರಯತ್ನ ಮಾಡಿ ಅನ್ನುವಂಥದ್ದನ್ನ ನಾವು ಹೇಳ್ತಾ ಇದ್ವಿ. ಆ ದಿಶೆಯಲ್ಲಿ ಡಾಕ್ಟರ್ ಮಾಧವಿ ಅವರು ಮತ್ತೆ ನಮ್ಮೆಲ್ಲ 4-5 ವಿಭಾಗಗಳ ಮುಖ್ಯಸ್ಥರುಗಳು ಆ ಒಂದು ಕಾರ್ಯೋನ್ಮುಖರಾಗಿದ್ದಾರೆ. ಇದೇ ರೀತಿಯಾಗಿ ಶೈಕ್ಷಣಿಕ ಚಟುವಟಿಕೆಗಳು ನಡೆಯಲು ನಾಳೆ 24ನೇ ತಾರೀಕಿಗೆ ನಾನು ರಿಷಿಕೇಶ್ ದಾಮಳೆ ಅವ್ರನ್ನ ಕರೆದಿದ್ದೀನಿ. ರಿಷಿಕೇಶ್ ದಾಮಳೆ ಅವರು ಅಮೆರಿಕಾಗೆ ಹೋಗುವಂತವ್ರು. ನಾನು ನನ್ನ ಸೀನಿಯರ್ ಅವರು, ನಾನು ಬರಲೇಬೇಕು ಅನ್ನುವಂತ ಒಂದು ಮನವಿ ಮಾಡಿದಾಗ ಅವರು ಕೂಡ ಬರ್ತಾ ಇದ್ದಾರೆ. ಅವ್ರು ಸುಮಾರು ಒಂದು 3 to 4 ಅವರ್ಸ್ ಇಲ್ಲಿ ನಮ್ಮ ಕಾಲೇಜಲ್ಲಿ ಇರುವಂತದ್ದಾಗ್ತದೆ. ಸಾಕಷ್ಟು ಅಂತಾರಾಷ್ಟ್ರೀಯ ಮಟ್ಟದಲ್ಲಿ ನಡೀತಾ ಇರತಕ್ಕಂಥ ರಿಸರ್ಚ್ ಗಳ ಬಗ್ಗೆ ಸಾಕಷ್ಟು ಅಗಾಧವಾಗಿರ್ತಕ್ಕಂಥ ಜ್ಞಾನವನ್ನು ಹೊಂದಿರುವಂತರು. ಅಟ್ರಿಮೆಡ್ ಅನ್ನುವಂತ ಒಂದು ಕಂಪನಿಯನ್ನು ಸ್ಥಾಪಿಸಿ ಅಮೆರಿಕಾದಲ್ಲಿ ಆಯುರ್ವೇದದ ಒಂದು ಕ್ಷೇತ್ರಕ್ಕೆ ಕೊಡುಗೆಯನ್ನು ಕೊಡ್ತಾ ಇರತಕ್ಕಂಥ ಮಹಾನ್ ವ್ಯಕ್ತಿತ್ವ ಹೊಂದಿರುವಂತರು ರಿಷಿಕೇಶ್ ದಾಮಳೆ ಅವರು ನಮ್ಮ ಕಾಲೇಜಿನ ವಿದ್ಯಾರ್ಥಿ, ಅಂದ್ರೆ ಬೆಂಗಳೂರು ಕಾಲೇಜಿನ ವಿದ್ಯಾರ್ಥಿ. ಹೀಗಾಗಿ ಅವರ ಒಂದು ಸದುಪಯೋಗ ಪಡ್ಕೊಳ್ತಕ್ಕಂಥದ್ದು ಆಗ್ಲಿ ಅನ್ನುವಂತ ಮಾತನ್ನ ಹೇಳ್ತಾ ಈ ಒಂದು ಕಾರ್ಯಕ್ರಮ ಯಶಸ್ವಿಯಾಗಿ ನಡೀಲಿ. ಸಂತೋಷ್ ಬೆಳವಡಿಯವರು ಅಷ್ಟೊಂದು ಶ್ರಮಪಟ್ಟು ಬಂದಿದ್ದಾರೆ, ಅವರ ಜ್ಞಾನದ ವಿನಿಯೋಗ ನೀವೆಲ್ಲರೂ ಮಾಡ್ಕೊಳ್ಳಿ ಅಂತಕ್ಕಂಥ ಮಾತನ್ನ ಹೇಳ್ತಾ ನನಗೆ ಮಾತಾಡೋಕೆ ಅವಕಾಶ ಮಾಡಿಕೊಟ್ಟಿರ್ತಕ್ಕಂಥ ಎಲ್ಲ ವಿಭಾಗದ ಮುಖ್ಯಸ್ಥರಿಗೆ ಧನ್ಯವಾದ ಅರ್ಪಿಸಿ ನನ್ನ ಮಾತು ವಿರಾಮವನ್ನು ನೀಡ್ತೀನಿ.
Thank you so much, sir. I wholeheartedly welcome Dr. Rajshekar Ganika, HOD, Department of Dravya Guna on this occasion. I welcome you, sir. I am also very much delighted and privileged to welcome the in-charge principal, Dr. Shyam Kishore Singh, on this occasion. I wholeheartedly welcome you, sir. I am also very much delighted, privileged and honored to welcome HOD, Department of Biostatistics, Dr. Madhava Diggavi, on this occasion. I welcome you, sir. I am also very much happy to welcome all the professors, all the PG scholars on this occasion. I welcome you. Now I request Dr. Madhava Diggavi, sir, to speak few words regarding this session.
Respected in-charge principal of the college, Dr. Shyam Kishore Singh. Today's guest speaker, Dr. Santosh Bewadi sir, Dr. Rajshekar sir, all the teaching faculty members, and my dear first year PG scholars. Today we are here to have the guest lecture series for first year PG scholars. It is mandatory to have a certificate after participating in a workshop before you submit the synopsis. And another issue is we should have some different thoughts before we submit a synopsis. And if you do synopsis, this series of guest lectures or a workshop, when you just take admission, you will be raw hands. You will not understand much. So for that purpose, 2 or 3 months once we take class, then if we organize some workshops, you will be able to understand the subject properly. In that way, we are having 4 or 5 guest lecture series from different scholars who are expert in this field. Among them, today is the inauguration come first guest lecture series by Dr. Santosh Belwadiji. And many times, synopses are rejected or synopses are written with some recommendations before they submit. For that purpose, you must keenly understand the steps involved in submission of the synopsis. For that purpose, this topic has been taken. Okay, even though all of you will try very well to write a good synopsis, but still you have to remember few points. And I have requested Dr. Santosh Belwadiji to see that the students will understand and they will have interaction, questions and answers, and see that nobody's synopsis will be rejected. Or many comments or many recommendations will come in the approval committee. Whichever the process goes in Rajiv Gandhi University, that is well aware for Dr. Santosh Belwadiji. So I am thankful to our principal ma'am who has given always a supporting hand and blessings to organize such activities. And I am also thankful to all 5 PG department HODs and their faculty members who are supporting our first year PG scholars to participate and encourage them. And I am thankful to all PG first year scholars who have taken keen interest in organize. Any mistakes are there, we will learn from that and we'll correct it in the next coming guest lecture series. And with these few words, I wish this workshop to be very successful, meaningful, and understandable, and it will improve your moral boosting, and also it will give you some courage and confidence to submit the synopsis. With these few words, I am thankful to all PG scholars for organizing this event, and thank you.
Thank you, sir. Now I request Dr. Shyam Kishore Singh, sir, to speak a few words regarding this session.
On behalf of our beloved principal, Saida Fatima Madam, who is on official duty, I have taken up this charge as a principal for a few days. Sir, it's my privilege to welcome you on behalf of the institute. Thank you, sir. And when I'm just attending this, I remember my days when we used to do theses. If we also had such an opportunity to know what is synopsis, what is thesis, it would have been still a better, and probably would have done better theses and more presentations in a right way. I really feel jealous of after having seen this, such a great personality has come over here, and he is sitting in the midst of us, and really it is everybody, students and teachers' opportunity to make use of our beloved guest today. Sir, on to my dear students, I take this a privilege that not only the students are learning, even we are having an opportunity to have in midst of you, and so that we teachers will also learn from you, sir. Thank you very much, dear students, and thank you, dear organizers, for giving me this 2 minutes opportunity to talk before you. Thank you. Let us have the program commence.
Without much delay, we shall begin the session.
Thank you. All the best.
ಇಲ್ಲೇ ನಿಲ್ಬೇಕಾ ಇಷ್ಟು ಇಟ್ಕೊಂಡ್ಬಿಡಿ ಸರ್ ಇಷ್ಟಿಲ್ಲ ಸರ್ ಕರಿಬೋದು ಬ್ರೇಕ್ ಆಗುತ್ತೆ Hello, hello. Am I audible? Hello. No. Yes, sir.
अज्ञानतिमिरन्दस्य ज्ञानाञ्जनेन शलाकाय चक्षुरुन्मीलितेन तस्मै श्री गुरवे नमः. Morning to all. Respected President of today's function, in charge Principal, Dr. R.K. Singh sir, Senior Professor, my senior friend, Dr. Rashekar Gange sir, my senior friend, well-wisher, Dr. Madhu sir, teaching faculty who present over here, my friends, Dr. Kendall Bhatt, Dr. Shivling, Sampath Kumar, Dr. Rajesh sir, all busy scholars. I'm very much happy to be here. Before to start my presentation, I'll tell one secret of my first year UG in the year 1993. I got C first year BMS seat in Taranath Ayurveda Medical College in the year 1993. Nobody knows it. Only kept secret. I thought this is the right platform to put forth in front of you. I came over here, I did admission, and in the second round, I got Government Ayurveda Medical College, Mysore. So at this juncture, I wholeheartedly congratulate the principal of this esteemed institution, Dr. Fatima Madam, Dr. Rashekar Gange sir, Madhav Digami sir, all teaching faculty for upgrading this institute, one of the esteemed institution in Karnataka. I have seen what is the status of this institution in the year 1993. Totally, entirely different. There will not be a good and quality basic needs at that time. There will be heavy rain at that time, and in the office, I'm recollecting the year 1993. There will be a lot of waters in the office because of heavy rain and in Kattada Shitilata. In World War, we can say Kattada Shitilata. At that time, the status of some government colleges will be like that. But comparing to 3 decades, it is one of the highly infrastructured with experienced teacher. What we are seeing now, really, you are very proud to feel that in all the infrastructure with quality of teacher, you have selected a meritorious and you have entered post graduation in this esteemed institution. I congratulate for you and wishing you all the best. So I used to quote always one quotation: Satata Adhyanam Vadam Paratantravalokanam Tadvidya Sambhasho Buddhivardhananam Sreshtaha. So how to upgrade our knowledge? So always we should do regular study, Satata Adhyanam Vadam, selecting one topic and discussing pertaining to that. Paratantravalokanam, referring the contemporary science, nowadays it is very mandatory to upgrade our knowledge with the contemporary. Then only it will be acceptable by the other people or contemporary specialist. So these are all the criteria or the parameters to upgrade our knowledge. And the Taranatha Government Aeronautical College and all teaching faculty, including principal, has taken lead in organizing this workshop, particularly on synopsis writing, how to write and synopsis. This is need of our. So how to enhance our knowledge? So such interactive sessions is very mandatory to upgrade our science or upgrade ourselves. So I congratulate the department for organizing. Stand like this. Huh? You take one chair, otherwise you sit. Or is there any remote control you can use? Huh? It's very difficult to stand here up to 3 hours. You take a chair, if you don't have any objection. Remote ಇದ್ರೆ ಇಟ್ಕೊಂಡು ಅಲ್ಲೇ ಕೂತ್ಕೊಂಡು can change. So in my discussion and interaction, I have framed the discussion. The first one is some of the basic informations concerned to research methodology. The second point is what are the contents of a synopsis. Unless if you don't know what is the contents, and for each content or each title, what are the relevant informations is to be highlighted. If you have not followed, you will not write the good synopsis. So that's why in the second point, I'm highlighting about the contents of a general synopsis and what is to be highlighted for each title. And the third point, in particular, how to write a synopsis by taking certain examples. By taking certain examples, how we should write down the synopsis, what is to be concentrated like that. Some clinicals synopsis writing and very less about non-clinical writing. So there will not be much difference. The Rasashastra and the Dravyaguna usually select the experimental study or animal experimentations, and Rasashastra scholar used to go for experimental study, analytical study, phytochemical study like that. So instead of here, if you are carrying out some animal experimentation, in a clinical, we take the sample size. There, you will add albino rats if you are trying for animal experimentations. The sample size, how much animals you are taking. So like that, source where you are collecting everything. So the protocol or the structure will be same. Based on your speciality, mild changes will be there. So clinically, we are selecting certain investigations, certain objective parameters. So in that, they will not be like that. But source, conducting the studies and analytical studies will be followed in the recognized, well-established laboratory. So you have to specify there where you are conducting all these things. Next slide. So let us see what the basic information about the synopsis. Synopsis is a sketchy summary of the main points of an theory. So now you are at a stage to write an synopsis, which you have to carry out in the second year as a main research project, as a partial fulfillment of your post graduation. So how you are conducting your research project, you are giving a skeleton of that research project, that is nothing but synopsis. So we use dissertation, particularly for PGs or post graduation, and for PhD we use the term as thesis. So majorly, what I feel, the thesis will be little extensive than the dissertation, but in the United States, for both they use as a thesis or dissertations also. There will not be much difference in the meaning of thesis and dissertation. So thesis and dissertations are sometimes used interchangeably. So we can say we mix it. But as per Rajiv Gandhi University of Health Sciences, when we are a PG scholar, we will write the dissertation for the partial fulfillment. Here we are submitting the dissertation. Or when we are doing our PhD, so we will write as a thesis. Okay. So a dissertation is a long form piece of academic writing based on the original research conducted. So what is the dissertation? So you are selecting an area and you are preparing the skeletal of that and you are submitting in the first year and you will conduct actual your research, original research, and you will write down according to the content or according to the guidelines. So that is nothing but the dissertation. So we used to write MD or PhD, as I told. So interchangeably or in both contexts we used. But better when you are as a PhD scholar, write down dissertation, and if you are a PhD scholar, you can go for thesis. So that is only the simple difference. Coming to the contents of a synopsis. So you are writing at a stage, now you are writing at a synopsis. And what are the contents? General contents will be, let us see. And later, for each content, what are the points to be highlighted? That to relevant points, not what you will elaborate as far as possible, huge quantum. That is not necessary. It should be precise and more informative. For each content, it should use justice to your content. So, and at that pattern, you should go for this. So, these are all the contents need for the study, review of literature, hypothesis, previous research work done on the topic, what you have selected, which is similar, aims and objectives, materials and methods. Under materials and methods, various subheadings are there, like source. So in that source of patient source or subject source, drug source, literary source, diagnostic criteria, inclusion criteria, exclusion criteria, investigations, subjective parameter and objective parameter. So like that, there will be subheadings we'll discuss in detail in when we start in particular investigations. So this is also most important, not which are relevant to your study, not enlisting. So on what basis you are diagnosing, what specific investigations you are going to carry out. So we can bifurcate here as a diagnostic and as a screening. So under this setting, we have to write down assessment of results. So this is very important nowadays. So earlier, 5 years back, there will be a different pattern of writing dissertation and doing writing synopsis and doing researches. So nowadays, IEC very strictly observing everything, and if your work should be accepted by the contemporary science. So you should go for certain objective parameters. Keep it in mind. So whatever you select your synopsis area, research area, there should be objective parameter. Then only it is evidence-based practices or evidence-based researches, and everybody will accept. The contemporary science will accept, and your study will have more weightages. So that's why keep it in mind. Go for some objective parameters. That is very important. Overall assessment, ethical clearance. This is very important. You are legally accountable because you are doing human subjects. You are going to try over the human subject is very important, and even you are doing on the animals, that will be very accountable rather than the human beings. You cannot kill a simple albino rats. You don't have a rights to kill. But when you are doing your research for the wellness of the human beings or the society with the permission of IEC team, then only it will be permissible. Otherwise, you don't have a right. So, for to conduct your research legally or morally, the IEC approval is very much important. We'll discuss in the later, and list of reference and bibliography. So this is very important. Always when we submit the research synopsis, and after review, it is a common dialogue or a common question. Your bibliography or list of reference is not in Wencor style. So I'll write I'll tell what is the Wencor style and how your reference should be written. So it should be mandatory. At the end, last page, you are going to write down the list of references or bibliography. So that should be written under the heading of Engar style. I will give clarity later. Okay. So what is a preclinical study? So when you are doing research on a new medications, you should not directly advocate it on the human subjects. So it should be prior to that, it should be tried on the animals. That is called animal experimentation, and this is also known as a preclinical study. The new medications cannot be directly subjected over the human beings because you don't know what is the outcome of this, whether it is effective or non-effective. So to prove that, it is very mandatory to conduct this research on the animals. So that is called preclinical study. Animal experimentations. So certain new drugs, particularly the Dravana scholars used to study the drug toxicity, efficacy of the drugs. So we'll take some example like antipyretic, anti-inflammatory of certain single drugs. Even Rasa Vaidyas also used to study certain like Shodhara plus yoga as anti-inflammatory like that. So they have certain formulations. Based on their action, you will try to study whether which effect it is having like that. And analytical study, particularly the Dravya Guna, particularly Rasha Shastra, these scholars used to conduct this analytical study. So you are selecting certain formulations and you are analyzing the hardness of that medication, ash value, alcohol value, like that. So different aspects are there. We'll discuss in particular in the next slide. And clinical study is most important. So we are evaluating or you are advocating the medications on the human subjects, either it is in the form of Shamana Aushadhi or with a certain procedure in terms of Panchakarma or Shodhana, and evaluating its therapeutic effect based on medications on a specific condition or a specific disease. So that we call it as a clinical study. Next. As I listed, what are the contents of the synopsis? So let us see what do you mean by this. For each heading, what we have to write with the relevance for each heading. The first point is the need for the study. So what is the need for study? It is very important. The first question, if you are telling one title and medication, I'll ask what is the need for study. So you have to give justifications why you have selected in the similar way. Existence of the disease. Suppose if you have selected one conditions like Gridresi, so what is the existence of that particular condition? What are the contemporary contributions? What are the complications with that particular drugs? You have to highlight. Suppose I'll take one example, Gridresi, and we'll go for certain examinations, and in particular we'll go for MRI scan, and we'll get bulge protrusion of L4, L5, and S1. And the modern doctor used to write like certain neural pain depressor like gabapentin and some hydroxychloroquine painkillers, fluoridines, and some external gel to mask the pain. And continuous taking of this gabapentin and hydroxychloroquine, etc., will lead to the adverse effects on the body. So to overcome that, from the Ayurvedic point of view, what best we can do, how our Panchakarma, Shamanaushadhi, Rasayanas are helpful in relieving the conditions as a preventive, health promotive. That you have to give justifications. That is comes under need for study. Following? Following? Yes. Limitations, prevalence. Prevalence and incidence rate is very important when you are selecting your clinical your research protocol. So usually you write prevalence in the world population this much and this much, but you are not concentrating on the Indian population, and you are not concentrating on Karnataka population, and you will not concentrating on Ballari district population. So be specific. So there is certain formula, and usually we ask the question even in research methodology when we are presenting synopsis. So if I ask right now, I'll kept 40 sample size. Yes or no? If I ask what is your sample size, Now you are planning. You will tell 40 sample size. Next question, my question is, how you decided 40 sample size? Then you will keep on. University previous scholar did like that, sir, and university told 40 samples, but it is not a relevant answer. So there is a specific calculations of the sample size. So usually it is based on the prevalence rate and incidence rate is one point. The second point, in an academic year, that particular disease, as I told Grudrasi, in 1 year, how much of the patients are reported to our hospital? So based on that, we have to calculate. This is second point. So like that, there is one formula also. I will tell how to decide our sample size. Then Ayurveda contribution. As I told, we practice Amavachana, Kostashodhana, then Panchakarma, then Shamanaushadi. So how they will be helpful? So all you have to give justifications in the need for study. Statement for the problem. We call it as in a one word statement for the problem. So you have selected the problem. That is Grudrasi. Statement. What medications you have selected? How it will be helpful in relieving the problem. That statement problem, what we call in the heading, and you have in an explanation form from the Ayurvedic point of view. So if you discuss under this heading, that becomes here for the study of ideal informations. Review of literature. Review of literature. Suppose if you have taken Rudrasi, so start with the Vedic literature, then Brahmatrais, and 21st century authors. Rudrasi, if you are not getting direct Nidana, Lakshana, or Samprapti, Chikisa Sutra directly, but in the Charaka Chikisthana, 28th chapter, the Samanya Nidana of Vata Vyadhi, and in particular, the Chikisa Sutra for Gudrasi is explained in Charaka Samhita. You should write as it is and give one superscript number above the head number 1. This is called superscript. It is very mandatory. I'll give some examples. And number 1 at the last page, you should write what is number 1. Right? In style, you should write like Charaka Samhita, Kashinath, Shastri, Chakrapani, commentary, edition, publication, chapter number, verse number, page number. So this is the art of writing the bibliography or the referencing as per the Venkat style. So like that, wherever explained concern to your disease, your treatment modality, what you have to collect the information from various Samhita Grantha, write down what are the informations pertaining to your research is explained in different context and go on writing the numbers in the Venkat style. Superscript and you have to write at the end. Next. So just I told research question. And research question, this is very important. Many of the synopsis don't write this. Even I have evaluated the dissertations also from this institution also synopsis as well as last time dissertation. Most of the scholars neglect writing research question and hypothesis. So these are all very much necessary, which gives clues for selection of that topic. So research question, nothing but so you have selected some like example Trayodashan Gugulu, which consisting of a number of tracks, including Gugulu. So most of these dravyas consisting of Vatadara. So like Rasaguna Viri Vipaka, what majorly they are consisting of. Along with that, what are the pharmacological action and what are the active principles present in that yoga and how it will be helpful in relieving the Gudrasi condition or how it will be helpful to check the samprapti of the disease. So you have to view the picture. You have to view the picture, then you are framing the research question. Is Trayodashan Gugulu is effective in Gudrasi Vata? Is Paitarana Basti is effective in Gudrasi Vata? That is research question. So here also you have to concise and write down the problem statement. So based on this, I am framing the question, research question, and selected the area to do research. So that is research question. Next. Hypothesis. So this is very important. Unless if you are not preparing your hypothesis, you should not you will not produce further. That is very important. Before selecting or before finalizing, you should have a hypothesis. So, and hypothesis is nothing but a solution for the suggested problem. Simple. You'll read all these in research methodology. It is a solution for a suggested problem. And we represent this as H0, that is null hypothesis, and H1 as alternative hypothesis, and it is also called research hypothesis. So this is very important. If you have selected some yogas, null hypothesis you have to frame. Your treatment is effective or not. Like ತ್ರಯೋದಶಾಂಗ Google is not effective in Krudrasi. Vaitarnavasti is not effective in Krudrasi. H1, ತ್ರಯೋದಶಾಂಗ Google is effective than Krudrasi. This is alternative hypothesis or research hypothesis. So based on your research protocol, you have to build up or you have to state the research hypothesis. Next. Previous research work. Try to restrict or stick on to the study which will mimic to your study. Don't write n number of lists, 20, 30, 2 or 3 pages. It will cover previous study. Not necessary. Suppose if you are selecting one condition, Medovara Gugulu in Staulya or Medoroga, pertaining to this, that too 1 or 2, 3 years, maximum 5 years earlier. I have came across 1975, 1979, 1980 study has been carried out. Not necessary. At least 5 years before. Suppose we are writing right now 2021. Start with at least 2015 onwards, not before to that. If the research is not available, 1 or 2 before to that you can select. But make maximum up to 5. Don't expand, not necessary. Previous study related to your work, you have to list out. That is very important. Aims and objectives. So this is very important. Suppose if you are selecting Panduroga, you will write aims. Literary search on Panduroga. Need not n number of Panduroga, n number of Kamamata, n number of Sandhivata. Studies has been carried out. You are not to take all copy 100 page and pasting it. It is not main aims and objectives. So that's why keep it in mind. The primary objective is very important. Primary objective and secondary objective. I'll take one example to clarify what is primary objective and secondary. Primary objective. So we have Shula, Shota, Stabdata in Gudasi. To evaluate the effect of Vaitarana Basti, particularly on Shula and Shota, is the primary objective. And rest other clinical features will manage, that will comes under secondary objective. So this is primary objective and secondary objective. The synapses are upgrading. So we used to come in contact when we are discussing in the ethical committee. The contemporary doctors are visiting, and they will take some guidance from them, our side, and we will take some guidelines and upgrade points pertaining to our synapses. And we are guiding our scholars as a primary criteria, primary objective parameters and secondary objective parameters. So all I have discussing in this platform. Next. Aims and objectives. So primary outcome. Suppose I have kept as an objective parameter for Gridrasi, MRI is mandatory. Routine speak pura Petipusturu, Jano, Janga, Parakrama. Nowadays, no weightage for this. So you should work your Shamanaushadi and Basti Chikisa. What efficacy over L4, L5 bulge or the protrusion. Then only your research will be acceptable and it will carry more weightage. I'm not seeing all these specura whether it is reducing or not. I'll take MRI. When I got the impression as L4, L5 bulge protrusion, then I will consider for the inclusion criteria. I will give my medicine, Basti, I will give my Shamanaushadi, I will do follow, and after completion, I will go for one more MRI scan, and I will compare before treatment and after treatment, what changes I will claim. Definitely will get the results, and we will declare with evidences. Then only it will be acceptable. The contemporary science will be acceptable. Ayurveda are having more authenticated therapeutic effects we can claim. Not only subjective no value, no values, but you should work. I'll give some more other examples when we are discussing in particular of certain synapses. Next. Secondary objective, other clinical features. Changes over the other clinical features pertaining to illness. Materials and methods. So this is most important. What are the materials? What are the materials you have taken for your study, and how you are utilizing, how you are advocating, how you are implementing these things? You should discuss in particular. Follow. So in that, the first point is source. Source. So under this heading, three main points will be there. One is we earlier scholar and even teachers, guides also use patients. Patients. So rather than using patients, we should use better subjects. Better it is used for subjects. That is very ideal. So we should make three headings under the source. That is, one is subject source. The second one is drug source, and the third point is literary source. So under the patient source or the subject source, so from where all you are collecting the subjects for your research work. So you have to quote. For example, if you have selected a study, the diagnosed subjects of Vidrasivata will be selected from OPD, IPD of Taranath Ayurveda Medical College and Hospital, and special camp conducted for the purpose will be selected for the clinical trial or for your study is the patient source. Followed? Next, the second point is drug source. Drug source. So how you will state? You will select the medicines from the market. No, it is not sufficient. So whatever the ingredients available, the drugs, the raw drugs will be properly identified by the faculty of Dravaguna teacher and a botanist, and it will be purchased authenticated vendors. That is the statement in your drug source. And the medications will be prepared in Rasashastra and Vaishyaja Kalpana or pharmacy of TGAMC Bellary. So that is the drug source. And the process of medication or preparation of medicine. So this is mentioned in Vaishyaja Ratnavali of this Adhikara, and the medicines will be prepared as explained in this context. So you have to give the numbers superscript and you have to write at the end. These medicines will be prepared as per author or acharyas like this and prepared in our Rashashastra and Vaishyaja Kalpana pharmacy. So this is, what is this? Preparation of the medicine. Next is literary source. So literary source, how you are writing the literature pertaining to the disease from the Ayurvedic point of view and the contemporary science, and upgraded with the recent medical journals and relevant websites. So it is mandatory. There are different research portals. You know what is research portals? Research portals, we have a number of research portals where if you browse, suppose if you are selecting some kind of diseases, if you browse what are the latest research has been carried out, and it will guide or it gives some informations for your work. So you have to take some relevant publications, relevant websites, which will upgrade your synapses for the authentications. So it is mandatory to approach different research portals and take a relevant references, relevant publications that will be more authentications for your synapses. So you are collecting the Ayurvedic literature from the Ayurvedic classes, modern conditions from the modern text, and upgraded with the recent medical journals and relevant websites. So whatever you are using or collecting, you have to write at the end of your research, the Venn style or the bibliography. So don't write only www dot this like that. So you have to open that, and there you will get the date, the particular date when you have seen everything. The whole thing is to be enlisted in the bibliography, not only the website number www.pubmed.com. It is not sufficient. So if you open this site, you will get the date, everything, informations. The whole information is to be taken as a total reference, not only the website
Okay, so you should keep it in mind. So these are all the source concerned to source. So next is study design. Study design, it is also very important. We'll discuss what are the different study design here. Open labeled, randomized, control, or comparative or observation. So we come across different studies: observational study, comparative study, clinical study, huh? Control clinical study. So all these you will come across this. So what type of study you are selecting, what type of study design you have to quote. So open labeled means everybody knows. The patient will know, the research will know, your guide will be knowing what you are giving. Is open labeled. Single blind is patient don't know, the researcher will know what medications they are giving. Am I audible? That is single blind. Double blind, neither the patient nor the researcher. The guide will analyze. Next, triple blind, neither the patient nor the scholar or the researcher nor the guide. The fourth person will analyzing its efficacy. That is triple blind study. So these are all the different study design where we have to take into account for the selection of your study. That is very important. Sample size. This is very important. How many subjects you are considering for your clinical trial is very important. It is mandatory, as I told. So question arises. So you should know the border of these things. So on what basis you have selected the sample size? So no doubt every dissertation previous work 40 is fixed, but if you calculate based on your work, it is nearer to that. Huh? Even if you fix 40 size, you should able to answer how you have calculated or on what basis you are decided the sample size. I'll tell later how it should be calculated. Based on the prevalence, based on the population, based on the patients reporting to the hospital in a year. So all these to be taken into account to fix our sample size. Total number of patients or the subjects selected for the study and how many groups. So it comes under sample size and grouping. Study design, sample size, grouping. So totally we will selecting 40 patients and made into two groups. Group A consisting of 20 patients, Group B consisting of 20 patients. So you have to narrate in this manner. Inclusion criteria. So how you are including the patient, on what basis? On what basis you are selecting your patients for your clinical trial? You have to quote. You have to quote. That is very important. Next. Exclusion criteria. So why you are not including the patients for your condition or your trial? That you have to quote clearly. Signs and symptoms of Grudrasi, you will write like this only, but it is not sufficient. If you are doing Sandhivata, you have to narrate Janu Sandhi Shula, Shota, Stabdata. These are all the cardinal features for Sandhigata Vata. If you are, you have to write. Only clinical features of Sandhigata will be included, not sufficient. So you have to specify what are these clinical features, cardinal features, not if 10 clinical features are the signs and symptoms, need not to write down all the things. The Pratyatmaka Lakshana, cardinal features, what we say, you have to write down when the diagnosed patient or the patient complaining of signs and symptoms of Sandhigata Vata. Like this, you have to write down the cardinal features. Whatever you are selecting the condition, based on that, you have to narrate. Exclusion criteria. Suppose if you are taking administering Basti, inclusion criteria, you will write who is fit for Basti Chikisa, inclusion. So that's why here you are not including, because the patient may suffering with some erectile disorder. He may not be fit to take the Basti Chikisa. And some traumatic conditions are there. Some infectious diseases are there. So some other systemic diseases are there, which will interfere with your treatment, that will not be considering for your study. Clearly, you have to quote in the exclusion criteria. Next. Criteria for diagnosis. So based on signs and symptoms also we'll write, but better if you have some objective parameter to diagnose. That is very important. For example, if I want to study on Ardhita Vata, so as a subjective parameter, Mukha Vakrata Aspashtavaka, Praseka, Lala Srava. These are all some signs and symptoms as an objective parameter, but it is not sufficient. So we'll take as a diagnostic parameter as Bell's phenomena positive. When Bell's phenomena positive, if you add, it carries more weightage. That will be the diagnostic along with the signs and symptoms. If diagnostic Bell's phenomena positive, I will write down. I will take a or I will diagnose as a Ardhita Vata. If SLR positive, if Lassiju's lip tests are positive, with Spik Pura Katipustur Janu Janga Padakrama, I will diagnose as a Gridrasi Vata. So we should have certain diagnostic parameters, then it will carry more weightage. Posology. This is very important. This is, we call it as interventions also. So this you are narrating from step 1 to last. For example, you are initiating Amapachana. What is the Amapachana yoga? What is the dose? What is the Anupana? What is the Aushadha Sevana Kala? What is the total duration? If you are giving Koshta Shodhana, what is the quantity? What is the ideal time to advocate? If you are selecting Basti, what is the Basti pattern based on the Sankhya? How many Niroha? How many Anuvasana? At what time you are giving in terms of Purvakarma, Pradarkarma? What is the total duration? Shamana Ushadi, Shamana Yoga, 1 tablet in bracket, how much mg, how many days, what is the dose, what is the Anupana, what is the Aushadha Sevana Kala, total Amapachana days, Koshta Shodhana days, Basti days, Shamana Ushadi days, then total study duration. I'll give some charts like this based on that. Just I'm giving instruction that your posology should consisting of in this pattern. Are you following?
Yes, sir.
Okay. We'll proceed.
Yes.
Last, total study duration. So in this manner, I'll give charts for some example, other example in particular. Investigations. Suppose if you are taking Amla Pitta, you are taking Grahini. Is there any investigations to diagnose Amla Pitta, Grahini? No. So certain conditions where we practice only based on signs and symptoms as a subjective parameter. So better you select a disease where you can do with certain investigations, you can do particular diagnosis. For Ashmari, USG pelvis. For Asti Saushiria, bone mass density. For gouty arthritis, uric acid. If you have any specific investigations, write down as a diagnostic. Even in the second point, you can go for screening. For example, to rule out some infectious disease, you can add CBC, and to rule out the diabetes mellitus, you can go for simple RBS as a screening, but not in general or not as a diagnosis for your disease. There is no role absolute. So that's why you should be specific. If you are screening, write down under the heading of screening. If it is a diagnostic, write under the heading of diagnostic investigations. Followed? Assessment criteria. So we have subjective and objective parameters. So as I told in the earlier works, majority are the only subjective parameters. If a patient comes with Urodaha, Udaradaha, Amlodgara, Shirashula, we'll diagnose as Amla Pitta. 15 days, 1 month, we have given medicine. Urodaha reduced, Udaradaha reduced, Amlodgara reduced. On what basis you are claiming? Based on the patient's version. But such studies will not carry more weightage. So that's why you should have certain parameters. So earlier days, there will not be developed technology. So we should keep it in mind always with all Ayurvedic tools and with relevant modern investigation techniques, examination techniques, one should diagnose the condition and we should manage from the Ayurvedic point of view. So in the contemporary science, for different conditions, n number of different scales has been developed. So if you are working on Sandhigata Vata, OMAC scale is there, VAS scale is there. If you are working on Ardhita Vata, facial disability index scale has been developed. FDI, facial disability index, and one more Brackman scale. This is a standard parameters where you can assess your conditions with a certain objective and subjective parameters. And if you select these things for your research based on different, if you select such things, it carries more weightage, and you will give, or you will claim, yes, your treatment is working this much. Based on subjective parameters, you cannot. So try to work out, try to search, read, write, browse. There are n number of different skills has been developed from different authorities, WHO guidelines. Different committees has been developed, and they are framing different the scales to assess or to evaluate the efficacies based on certain objective parameters. You have to little work out. Definitely you will get. Next. So we'll also objective parameter with certain investigations also. Certain investigations, if you are analyzing iron deficiency anemia with certain medications, BT80 of iron deficiency anemia, or total blood smear, like that. And if you want to analyze your efficacy on total semen parameter before treatment and after treatment, you can collect. So like this, if you are selecting, this is must for your diagnosis, that is very good. This is very must. Investigation. After 1 month or 90 days of your treatment, once again that investigations, and you will compare. And you will give the justifications. So this is called evidence-based researches also. Think on this. Everything guide will not give you. Everything guide will not dump in front of you. The interest, how you are approaching. We are showing the guidance like this. You refer this, you refer this, you refer this. Will not bring everything to you, because you are the researcher. You should have that eager to read, write, discuss. Definitely you will give justice to that. And think what you are writing. Once, twice, discuss. Because once if it is approved, you cannot change the protocol. So that's why what you are giving, what medications you are selecting, what are the ingredients, whether gold is there or not. If gold is there, then who will bear the expenditure? Suvarna yogas. And some rare medications are there, herbs are there, whether they are easily available or not, whether it is easily preferable, what is the duration, how much quantity, what are the cost effectiveness, the method of administration, palatability, storage, everything, shelf life. So all these should be kept in mind before planning your research protocol. Assessment of results, as I told, how you are going to assess and what are the statistical tests applied. So earlier synopsis, what the scholar used to write, it is statistically analyzed by using relevant statistical test. Nowadays, relevant statistical test is not ideal. You have to quote. So how you should write, it is based on the sample size and based on the groups what you are selecting. So up to 30 sample size, there is one particular statistical test, and more than 40 sample size and 2 groups, there is a particular statistical test. If it is more than 3 groups, irrespective of the sample size, the statistical test is different. And how you are selecting the patient is also counts. If you are having 2 groups or 3 groups, you have to state how you are going to selecting the patient. Anybody? How you plan to select a patient? Suppose if you think 2 groups, or one, you may think 3 groups. How you are selecting a patient for group A, group B, group C? Anybody you can tell. If it is wrong, no problem. Nobody is perfect. Please stand up and tell, let other comes to know. Please stand up. If it is 2 groups, how you allocate? Don't hesitate. It is friendly discussion. This should not be. Whenever there will be a question, you should stand up and tell. PG means not sitting in a 4-wall room, transferring. No. Various platforms to read, write, discuss, exhibit. In a classroom, in a OPD, in a IPD, in a seminar, IDM, outside presentations. These are all the skills where you have to develop. Anybody, how you are allocating the cases? Suppose if you are taking 2 groups, how you are going to allocate the patients? Henry, be bold. It's a friendly discussion. If it is wrong, no problem. I don't mind. I will guide you once again. Yes.
So one group only with eye-centric intervention.
Huh.
In the other group, eye-medic as well as motor intervention.
Okay. If a patient number 3, third patient comes to you, to which group you will allocate?
So it is random.
Means
Selection.
random means
So if there are 100 persons in a group, so randomly we will select.
sit down, please. So one technique is please.
Open blinded will explain. Open blinded ಅಂದ್ರೆ ಅವ್ರಿಗೆ modern ಇದೆ ಈ ತರ.
ಇಲ್ಲ ಮೊದಲು ನಾವು ಎರಡು ಗ್ರೂಪಿನ ಆಯುರ್ವೇದ ತೊಗೊಂಡ್ಬಿಡೋಣ. ಕ್ಲಮ್ಸಿ ಮಾಡ್ಕೊಳ್ಳೋದು ಬೇಡ. ವಿಲ್ ಟೇಕ್ 2 groups Ayurvedic intervention. 2 groups are there. How you are allocating the patients?
Will explain.
It is bias. It is unbiased. Kudri, please sit down. Your selection should be unbiased. ಈ patient ಗೆ ಬಹಳ severity ಐತಿ, ಇದ group ಗೆ ಹಾಕು. This patient is having more severity. Group A treatment is very good. Keep it in group B. Group A. This patient very mild. Keep it in group B. This patient is having long lasting history. Keep it in group A. Why? Group A is very good treatment. This is called bias. In 1 minute, in research, it should be unbiased. I used to tell my scholar, unbiased, be optimistic. मेरा ट्रीटमेंट बहुत अच्छा है, इफेक्ट आने का है, ये ऐसा नहीं. No predetermined. What you are advocating, what you are observing, what you are documenting, either it is a positive or a negative, write as it is and claim in the conclusion. मेरा मेडिसिन बहुत अच्छा है, मैंने प्रिपेयर किया है, मैं किए सो बहुत अच्छा. This is called predetermined and ऐसा ही होना है, नहीं होता है ऐसा ही. This is not called research. Be optimistic. What you are observing, you write as it is. Don't allocate like this. We call it as a simple random technique. And another one, the computer generating allocations, what we call computer generating. For example, I'll tell we have 2 groups, 100 sample size, or 3 groups, 140 sample size. So there is one, if you browse Google, computation allocation, we call it as 120 sample size, 3 groups. If you allot and okay, if you give okay, it will display group A number, group B number, group C numbers. Suppose you are taking 17th patient. 17th patient, you have to look 70 number in A, B or C. Following? 17th number, 17th patient, either it is there in A or B or in C, because it is computer allotting. It is called unbiased. If it may be in A, it may be in B, it may be in C. So where it is showing, you should take that patient to that group. Following? This is reality of allocation of the group, selection of the patient. Simple random sampling technique we used to call. But this question will be asked, so you have to use some standard allocations, some computer allocation technique methods, like that. Not based on the severity, not group A is effective, group B is effective, not like that. So based on that you have to. So this is called unbiased sampling technique method. There are a number of you will read in research methodology. So this is the pattern how we are selecting the patient sample techniques, sampling techniques. This is called. So you have to allocate like this. Next. Overall assessment next. Ethical clearance. So this is very important for each research. You cannot do how you want the research, because you are doing your treatment for the human subject, on the human. It should not be. Everything is accountable. Everything is your research, what you have selected will be helpful to the mankind. So after proper justification, after relevant interactions, they will give the approvals. Either it may be the human or even in the animal experimentation. Even for animal experimentations also, you should project human ethical clearance committee approval. That is very mandatory. So it means everything is accountable. So you should do your research work morally or legally. So what happens? Have you heard Helsinki? Helsinki. So in ancient times or previous years, the researches were done to harm the human subjects or the society or the personal purpose they used to do. I will recollect some movies like Krish. Krish, they did some research and for the destroying the human, and one more movie. Vikram is the hero. I think he's I. So they are injecting some injection and made huh? They vanishes. So these are all some things. Why? To harmful to the others. These are also conducting some laboratory. You might be seen and recollect the movie. These some researches which will be harmful to the body or human subjects. So in earlier days, for the personal purpose, the different organs, the different body parts of the persons will be collected and they will do researches, and they are taking the life of the peoples. So that's why Helsinki has declared, that's why it is called Helsinki's declarations, and he framed certain guidelines to conduct the research legally. That is why it is mandatory to put forth your research protocol in front of the IEC member. After approval, it should be permitted for the registration in the university, and after completion of your first year, you have to initiate your research. Next. Superscript, as I told, when you are writing one sentence and taken it from Charaka Samhita, just you have to write down the number 11 and 11 number at the end. What are the reference, which reference number, chapter number, verse number, page number. So you have to write down everything. That we call it as superscript. Next slide. Washout period, or what do you mean by washout period? Suppose if you are taking some medications, already the patient is taking some medications, the question will be asked what you are doing for that. You are following what I'm telling? A patient is already taking some medications and you want to take for your clinical trial. So the question will be asked, next day you are initiating your treatment, or what you are doing. So there will be a washout period. So we should give some gap, withdrawing the medicines and give some gap. After this washout period, you are initiating your medications. So that what we call washout periods. Next, there is no particular fixed washout periods for a specific conditions. Okay, so this is take a snap. How to calculate the sample size? There is one formula, and even if you are not following, just you consult your statistician. They will guide you. Even in first year also, even in final year also, when you are presenting your dissertation, and even in synopsis, the most common question will be asked how you are going to calculating your synopsis, the sample size. So this is one of the formula. Next slide. This is the list of the references how you are writing by following Venkat style. So this is one of the things what you have to write: name of the author and editor, name of the textbook, sthana, chapter, verse, edition, publication, page number. Even in, I think, research methodology, there is one syllabus point, the art of writing the referencing. So Venkat style, Harvard, different things are there. Our Rajiv Gandhi University is following Venkat style. So this is the pattern. And here I have taken one example, how to write a reference in the form of Venkat style. Pandita Parashurama Shastri Sharangadhara, edited by Vidyasagar Sharangadhara Samhita, with Adamalla Deepika and Gudartha Deepika. Pradhama Kanda, 7th chapter, 106th verse, 5th edition, 2002. Chaukamba, Oriental Varanasi, page number 103 to 104. So this is the pattern of writing one example of bibliography or referencing. Okay. So this is one of the point just I finished. Pertaining to this, any questions you can ask, or even at the end also you can discuss, no problem. Okay. Any questions? Are you following my? Any doubts you can ask right now also, or you can write down. At the end also we'll have some interactions. No problem. Shall you proceed? Proceed. So this is most important when you are writing your synopsis. Some of the basic informations pertaining to research is very mandatory. So unless if you are not well versed with this, it is very difficult, or sometimes wrong interpretations may be possible. So that's why I am giving some of the hints or outlook of some important aspect of research concern. So n number of research definitions are there in the research methodology text. So I used to quote only this definition, validating the old facts with the present knowledge. I used to quote my scholar or take the definition, validating the old facts with the present knowledge. Acharya Charaka, Shushata Vagbata, during their period, they have tried these medications and clinically observed, and they are written. They were written in the text. But in the present knowledge, how we have to evaluating? We are taking the same things and evaluating right now, re-establishing the old facts, re-establishing the old facts with the present knowledge. How we are re-establishing? Taking the Charaka's version or Shushrutas or Vagbata, what we require in the present day, we are selecting and evaluating its efficacy once again with certain upgraded knowledge. For example, in Panduroga Chikitsa, Navayasa Loha has been highlighted by Acharya Charaka. What they claimed in the text, Alparaktaha, Alpamamsaha, Nissaraha, Shatilendriyaha, Vaivaranaha, like that they have mentioned. And they have given Navayasa Loha and they have proved all the signs and symptoms got relieved, and Acharyas they came, yes, it is very efficacy in Panduroga. What is our duty right now? Re-establishing the Navayasa Loha in Panduroga. Panduroga is a general. We have a different category. In contemporary, different anemia, iron deficiency, nutritional deficiency, ferritin deficiency, n number of classifications are there. So my verse is, in which type of anemia is helpful this Navayasa Loha? We do blood smear, either it may be we get IDA or ferritin deficiency, nutrition deficiency. We take certain 40 sample size and we'll give Navayasa Loha for 60 days and evaluating its effects. Yes, Navayasa Loha is highly efficacy in ferritin deficiency, or it is very efficacy in iron deficiency anemia based on the investigations. In ancient time, Alparakta, Alpamamsa, based on this, they claimed it is improved. Now we are claiming with evidences. This is research, re-establishing the old facts. Navayasa Loha is helpful in Panduroga. Is old facts. Navayasa Loha is effective in iron deficiency anemia or ferritin deficiency anemia. Following what I am telling? So this is the definition of research, evaluating the old facts, revalidating the old facts with the present knowledge. During that period, there will not be much developed technology. Morning, I have seen with Madhav Digami sir, your research laboratory, particularly the semen parameter analysis in the this institution. Nowhere in India, what I thought, nowhere in India, such facilities are available. This is one of the technology, Sukrakshaya, Klaibya, Shukra Dosha, Spatikabam, Dravam, Snigdam, Maduram, Madhugandiche. We'll claim, yes, Shuddha Shukra. Fenila, Tantu, Durgandhi, what we will claim? Shukra Dosha. Yes, how you will claim? You will evaluating this salmon in the microscope with a sophisticated infrastructure, with a sophisticated aid, and with photocopy you are evaluating. Huh? Yes or no? This is research. Yasya bija bhaga vaiva upatapto bhavati tasya anga vaiva upajayati. The defects will be in head, neck, tail. How? You will visualize with the sophisticated instrumentation, and you are claiming, yes, with photocopy, with its structure. Drug analysis, if you use it pharmacy college, if you use it KLE, if you use it SDM, drug analysis, at what time Yogaraja Google will disintegrate in your system? You can see we are creating the environment, we are putting the tablet and we are stirring it. At what time it will dissolve? That is disintegration of that medications with a specific time. That is the research from Rasha Shastra. Disintegration of that the yoga with the instrumentation. This is research. Following what I'm telling? Yes or no? Okay. Next. What is the methodology? Research is different. Research methodology. The techniques or the methods which you are following to conduct that research is called methodology. Research, revalidating the old facts with the present knowledge. Research methodology, the techniques or the methods what you are following to conduct that research is called research methodology. Next. We get so many, when you read a different research methodology text, the equivalent terminologies or some synonyms we can consider as invention, inquiry, exploration, verification, investigation. So a number of equivalent terminologies we come, the meaning will be to search the knowledge or revalidating. So all the equivalent terminology we can consider as under the heading of research. So this is very important. When you are planning to write a synopsis, you should keep it in mind. One is identifying the problem and collecting the information and formulating the hypothesis. Now you are at a stage. What is your main aim is right now? You have to identify the problem or selecting the problem. Next, after selection, you will go through all the classics and collecting the literature pertaining to that. Then you are extracting particular set of treatment for the selected problem. Then your problem is finished, your solution is finalized. The third point is you want to formulate your hypothesis. Following? No voice? Yes, okay. I will not leave another 2 to 3 hours. Okay. Now you are at a stage, selecting the problem, collection of the literature, formulating the hypothesis. Now you are at a stage. As I told, H0 null hypothesis, H1 is alternative hypothesis or research hypothesis. So when you are selected or you are finalized your research protocol, you have to frame null hypothesis and the alternative hypothesis. Next. After formulating your null hypothesis and alternative hypothesis, at the end of your research work, you have to statically analyze, and based on the result, you have to evaluate your hypothesis. If it is p-value is significant, you are claiming your hypothesis is your research hypothesis. If your p-value is not significant, you are rejecting your hypothesis. It means your medication is not effective in this condition. Followed? Characteristics of researcher. Aims to find out new facts, logical and objective orientation. Suppose you are concentrated, you are doing your research. So always research should be not targeting on one point, because the research has been carried out based on the personal observation. When you are doing some works or some research, you will have some new ideologies or new observations. We call it as chance observation. What chance observation? You are trying to look or find out something and you will found something. That is the chance observation, and that chance observations can be selected for the next research protocols also. So that's why researchers will be selected based on the personal observations. your teacher used to tell, yes, you take this protocol. So I have given many number of treatments to many cases, and I found this. So we'll do this research on the large sample size. This is called personal observations. So always you should be broad-minded when you are doing your research work. Based on personal observations, experiments, experts in research field, quest for answers, and the act of searching for something new, that is also research, requires a lot of patience. You have decided to give your treatment for 60 days, you cannot expect after 15 days or 30 days. Followed? So there should be a lot of patience and maintain records and reports. That is very important, where the people will lacking. I used to tell I did 1,000 patients of this and this. If you tell me, sir, show some evidences. If I tell, no, I don't have, can you believe me? No. This is the lacuna from the Ayurvedic point of view. That's why keep it in mind. Document not only your research protocol. So when you joined here, I observe n number of different treatments going on here. Challenging cases are coming here to take the treatment. Your teachers are guiding you. Yes, you do follow. You give panchakarma this. You do shamanaushadhi this. You give some rasaushadhis, some rasayana, some vajikarana. You note down the history. You document the investigation. You take the history of the patient, document it. After 1 month of the follow, you do documentation. Take the after treatment investigation. Take the patient's view. Take the treatment procedures before treatment, after treatment, after follow. Note down your points. You have a logbook. Yes. What you observe new thing, you have to return. You should write down. You should keep with you for your future, because everything is not you are recollecting when you require. But when you are documenting this, whenever necessary, these things you can refer this. So n number of treatments, n number of miracle results we are observing, but you are not documenting. Even you have good number of 108 megapixel camera, you are not documenting a single patient, and night duty you are doing like this. Yes or no? You are not followed? Not followed. Followed. Why I am telling, for other utilization you are using your mobile. Document for your trial version, observations, patients. When such platforms you present it, you will have good recognition. Confidence level is different. When you document this, when you present in front of your teacher, when you present in national and international, that gives credits to you, your guide, your institution. You should do it. You should utilize all these developed infrastructure instrumentations. N number of, but nobody is caring. Patient ತನ್ನ ಪಾಡಿಗೆ ಅವನು ಟ್ರೀಟ್ಮೆಂಟ್ ಮಾಡ್ಕೊಂಡು ಹೋಗ್ತಾನ. ಮ್ಯಾಸ್ಯೂರ್ ಮತ್ತು ಪೇಷಂಟ್ ಕನ್ಸರ್ನ್ಡ್ ಟೀಚರ್. The responsibility is only for the massive concerned teacher and patient. No scholar. We call it as in Kannada, ಕೆರೆದಷ್ಟು ತುರಿಸಿಕೋ. ಇಷ್ಟೇ ಸರಿ, ಈ ಕಡೆ ತುರಿ ಅಂದ ಎಲ್ಲಿ ಇಷ್ಟೇ ತುರಿಸೋದು, ಬೇರೆ ಕಡೆ ಬಾಳ ತುರಿಸ್ಕೊಂಡ್ರೆ ಗಾಯ ಆಗ್ತದೆ. ಹೌದಲ್ರಿ? No, should be always, because you are very lucky to be here. N number of patients are there. You will have good exposure in all the corners of this. Adilla, idilla, no facility like that. Don't tell this. What is available, you look in that vision and you can grab the knowledge. You have a lot of exposure here. See the things, follow the things, do the things, document the things, write down the article, write down the presentation. IDM, interdepartmental meet will be going on here, I think. A clinical student should document a case and present it in front of everybody, under the guidance of Digay sir, under the guidance of Bellam sir. Yes, OPD patient like this complaints, and these are the set of the treatment, and before treatment like this, and after treatment like this. That credits goes to you, your guide, your department, and let other 30 students will write down this treatment and they will practice when such patients will comes to their clinics. This is need of our. Why I am stressing here is, orally we will tell, I did 1,000 cases. I have not documented a single case. How will accept you and the contemporary science? Nobody will accept you. So try to document, and whenever they are required, try to exhibit. Thanks. Quality of researcher, logical thinking, highest integrity, should spend long time, sincere to his work or her work, optimistic, be always positive, don't fabricate, don't modify, don't go shortcut routes. Followed? Next. Anusandhana, parallelly, not necessary here. Next. Yes. Next. Next. So like that research we have, when you read Ayurvedic text, we come across a different equivalent terminologies for Anusandhana, like Anusandhana, Anveshani, Gaveshani, huh? Then Nirikshana, Parishodhana, Punarvimarsha, Punarnirikshana, Anviksha. So these are all the different terms come across in Samhita Grantha. You can consider as a nearer term or equivalent term, not directly as a synonyms. Equivalent terminologies we can consider for the Anusandhana. Next. So qualities of researcher. I used to quote this research, the quotation explained in Charaka Samhita, Sutrasthana 9th chapter, like Vidya, Vitarka, Vijnanam, Smriti Tat Partakriya, Yasha Shadgunasya na Sadhyamiti Nivartati. The researcher should all these 6 characteristic features. Vidya, knowledge. So whatever you read the Samhita Grantha, you should have all the knowledge. Vitarka, you should have always reasoning. Why, when, how. So you have to reasoning and find out the answer or the solution for this. Vijnanam, scientific method. So as I told, only based on subjective parameter we are cleaning, but we are not following any objective parameter. We are not taking any investigations. So always your approach should be scientific. That is very important. Smriti. Smriti is memory or Smarana Shakti. So you read all the Samhita Grantha, and whenever these are all necessary, you should recollect and reproduce that and utilize. That's why the Smriti is very much important. Tatparataha, repeated observation. So when you feel confidence, you should do yourself and observe. Either it may be in Panchakarma, either in OPD, either in IPD, or in the formulation of your products or the procedure. So when you do this repeatedly, when you observe, you will have good confidence. That's why repeated observation is very important. Then Kriya, practical knowledge. Unless if you are not doing massage, unless if you are not doing basti, you don't have much ideology. So first, the efficacy of Ayurveda, you should consume all Ayurveda medicine. To understand the procedures, you do yourself. Either it may be Dravaguna, Rasashastra, Panchakarma, Kayajiksa. You should involve and you should do the things. Then you will have good practical knowledge. So all these should be incorporated by a researcher or a student or a scholar or even teacher also. So as I told, there are different clinical study as open labeled, single blind, double blind, triple blind. So the same things as I quoted next. Open blind, everybody knows what treatments are going on. Single, the patient don't know and rest others. Double blind, the researcher and the patient will not, or the subject will not knowing the medication. And triple blind, all 3 person will not knowing, and the post researcher or chief or head of the section or authority will evaluating its efficacy. Next. Single case, as I told. So if you want to do a new case came to your OPD and a set of treatment will be given on a single patient and documenting, and that is called a single case. Rare conditions can be selected and documented, even it can be presented or even it can be published, a case report on this condition. That will be more ideal. It is called single design. The sample size is very minimum. Next. Case control study. So here there will be one standard group and one new group. It is called standard group and test group. So even you can select such treatments in your research protocol. One already proved, you can go for one group, and one new group which is said in the similar condition or the problem, you can take a new drug as a test. So it is called control clinical study. Okay, and next one is, next slide, control group. So even you can go for one Shamana Aushadhi standard, some the placebo also you can administer, even the role of placebo or even the role of mental status you can analyze with the placebo study also. Next. Next. As I told, study design, single, double blind, control, up to 40 sample size, one group, two groups. So based on that, you have to decide which study design you are undertaking. So as I told, the statistical test calculation, t-test when the sample size is up to 30 sample. So we used to calculate with the t-test, and paired and unpaired t-test when two groups and up to 40 sample size, we will calculate a paired test calculation within the group. Within the group A, patient number 1 before treatment and after treatment is within the group. Between the group A, patient number 1 before treatment, and group B, patient number 1 after treatment. So this is between the groups. Unpaired t-test calculation between the two groups, group A and group B, subject on subjective and objective parameter. Next slide. Z test calculation between two groups when sample size is more than 30 subjects. F test, ANOVA test calculation when more than two groups irrespective of sample size. Chi-square test, the calculation between the observed value as that of the expected value. And we have nowadays statistical software like Minitab and updated versions also. So why I am telling, these are all not much necessary for your synopsis. Only if your sample size is 40 sample size, two groups you used to practice or state is as paired and unpaired t-test. It is finished. So in PhD, we will take more than 100 sample size and three groups. ANOVA test, there we follow ANOVA test. But as a PhD scholar, we used to two groups when sample size is 40, paired and unpaired t-test. So just for a knowledge sake, even you have to know this in the research methodology also. Just thought of to give this statistical software. So if you have some basic ideology, so you have to upload your data by using this Minitab, and automatically it will calculate and give the reports. So even you can state with the statistical data you are going to calculate. So updated versions are there. Based on that, you can state which version you are using to calculate. Preclinical study, as I told, when you are thought of selecting a new drug, so you should not be directed on the human subject. It should be tried on the animal experimentations. After proving its efficacy, then one should go for human subjects. So that's why prior to that, animal experimentation is mandatory. That is called preclinical study. Next. Pilot study. Suppose your synopsis is approved, and even in ethical nowadays, the contemporary, the modern doctor, why you have selected, and any pilot study you have carried out, it is mandatory. So when you have plenty of time, you have to do. So otherwise, after approving of your synopsis, in between also you have to do minimum 6 sample size. So what is the purpose of doing this pilot study? It is more important. Even in your research methodology, these are all some of the syllabus points pertaining to you. So to undertake to check the feasibility and to improve the research design. You have to do pilot study. So for pilot study, minimum 6 sample size is mandatory. So it will be helpful to upgrade your actual research work. So that's why before conducting your main work, you should go for pilot study. It is mandatory. Next. Next. So as I told you, what is hypothesis, null hypothesis, and alternative hypothesis. This is very important. Next slide. Phases of clinical study. So we have 4 phases. So keep it in mind, based on the sample size, we can justify what is clinical phases, study phases. So there will be 4 phases: first phase, second phase, third phase, and fourth phase. So in the first phase of clinical study, the sample size is 20 to 80. In the first phase of clinical study, the sample size will be 20 to 80, and what we are doing, the safety dosage and identifying its side effects. We are going to study on 20 to 80 sample size, particularly the dose and identifying its side effects. That is phase 1. Phase 2, the sample size will be 100 to 300, and we are confirming the effectiveness of and safetiness of that drug in the second phase of clinical study. In the third phase, the sample size will be 1000 to 3000. That is the third phase of clinical study, and we are evaluating its efficacy and monitoring the side effect. And the fourth phase of clinical study, post marketing the medications. So after viewing its effects over this sample size, the product will be releasing for the market purpose. So always keep it in mind. Even for syllabus also, phases of clinical study will be there in your research methodology. Keep it in mind, the sample size, and based on that you can easily describe phase 1, phase 2, phase 3. Can anybody tell what is the sample size of first phase? Second? Third? Yes, very good. So as I quoted, how to write Venka's type. Same. Evidence-based research, I would like to underline this. So unless if you don't have any evidences that your medications worked out, then there is no acceptable, no weightage for this. So at least you should have some parameter, objective parameters based on hematological, radiological, sonological, some standard scales are the scores are some questionnaires. So anything pertaining to your study or what you are selecting for your research proposal, you should find out any of these things. If you follow before treatment and after treatment, it carries more weightage and you can claim the evidence-based researches. Next. So just I have given some examples. If you're selecting the Pakshagata, CT MRI, Basti Chikitsa is highly effective. What changes you can notice before treatment, after treatment, with some follow-up durations you can claim based on the scans. Next. So Manyastamba, Grivastamba, cervical spondylitis, MRI in case of cervical bulge and protrusion with Nasya Chikitsa, Bahya Chikitsa, Shamanaushadis, Rasayanas with proper durations before treatment and after treatment, you can tell some evidence-based changes pertaining to your research. As I told, Grudrasi, effective MRI in the bulge and protrusion, and in Arditavata, CT brain very rarely, but based on certain standard scales, you can claim the changes in the Arditavata, osteoarthritis, X-ray, BMD, VAS, OMAC scale. So based on these are all the some objective parameters to evaluate its efficacy. Next. Kampavata, MRI, CT, dopamine activity in brain. So based on this, Alzheimer's evaluating the Rasayana effect on particularly vitamin B12, like that. Avabhavaka, Projan shoulder, the efficacy will be on a particular objective parameter like nerve conduction test or sometimes X-ray, and inflammatory disease, particularly efficacy of your treatment on C-reactive protein or CRP, what we call. Some of the areas where you can think of some research to opt or to select. Most commonly we come across the patients Madhumeha. We have a number of Rasayana efficacies, Panchakarma, Shamanaushadis. Better if you take some objective parameters like HbA1c, glucose tolerance test before treatment and after treatment, you can give some evidence-based researches for Madhumeha. Staulya or Medoruga, Raktachapadikata, Twakvikara, particularly Ekapushta, Vicharchika, Shitapitta, Visarpa, Vatavadi in particular, Grudrasi, Avavaka, Manyastamba, Sandigatavata, Anukta Vatavadi, repeated stress injury. Why? We are using repeatedly laptop, computer, mobile. The patient used to come with pain and swelling in the upper limbs, particularly the fingers. So this we call it as Anukta Vatavadi. You can consider it Anukta Vatavadi and repeated stress injury, where our Shamanaushadhi, Rasayana, Panchakarma, Abhyanga, Sweda works very wonderfully, and you can select such things. Rasayana, particularly on Jara, Swasa, Panduroga, Annava, Sroto, Vikara, Vajikarana, under the heading of Kshira Shukra, erectile dysfunction, premature ejaculation. Hypothyroidism, most commonly we are getting such cases. You can select hyperlipidemia, gout steatitis, spine disorders, renal calculi, gallstone, Pittacia Ashmari, liver disorder, fatty liver, alcoholic liver disease, CKD, first stage, neuromuscular diseases, PCOD, male and female infertility, like Akshina Shukra. So these are all the area where we can plan very good results. Some concept-based areas, Shatkriya Kala, Amadosha, Vyadikshamatva, Swastha Panchakarma, Achara Sana, Pratimarshanasya, Prakriti assessment, Prakrita Shukra, Shukra Dosha, standardization of certain Panchakarma instrument, Snehapana procedures, Bahya Sweda Chikitsas, Bahya and Abhyantara Basti procedures, different Rakta Moksha instruments procedure. Not only evaluating the therapeutic effects, you can think of standardizations of procedures and standardization of certain instruments also you can thought of. This is excellent. So with this, we'll break 10 minutes and reassemble. Any questions you can now also you can ask, or at the end also, no problem. Next video. Good. In particular, outlook basics synopsis. ಆಮೇಲೆ ಟೈಟಲ್ಸ್ ಇದು ಒಂದು ಫುಲ್ ಇನ್ ಡೀಟೇಲ್ ಒಂದು ಸಿನಾಪ್ಸಿಸ್ ಒಂದು ಟಾಪಿಕ್ ಆಮೇಲೆ ಟೈಟಲ್ಸ್ ಏರಿಯಾ ಡಿಫ್ರೆಂಟ್ ಆಯ್ತು ವರ್ಚುವಲ್ ಆಯ್ತು 2 days ಅದು ಗೌರ್ಮೆಂಟ್ ಹೇಳಿದ್ದು ಅದು 2 ದಿವಸ ಅದು 2 days ನಮ್ಮದೇ ಪ್ರೈವೇಟ್ ಅಷ್ಟೇ ಪೆಂಡಿಂಗ್ ಬಜೆಟ್ ಜಿಗ್ನಲ್ ಸಭೆಯನ್ನು ನಮ್ಮದೇ ಮಾಡಬೇಕು ನಾವು ರಿಕ್ವೆಸ್ಟ್ ಮಾಡಬೇಕು ಎಲ್ಲ ಮುಂದುವರೆದು ಇದು
ಅದು ಲಿಂಕ್ ಇದ್ರೆ ಕಳ್ಸಿ ನಮ್ಮ ಕಡೆ ನನ್ನ ಲಿಂಕ್ ಐತಲ್ಲಿ
ಸೇವ್ ಮಾಡ್ಕೊಡ್ರಿ ಇದರಲ್ಲಿ ಒಂದು ಸೇವ್
ಮತ್ತೆ ಇನ್ಫೆಕ್ಷನ್ ಏನು ವೈಂಡ್ ಅಪ್ ಮಾಡ್ಬಿಡ್ತು ಮತ್ತೆ ಅವರು ಬಿಟ್ರೆ ಇಂಟ್ರೆಸ್ಟ್ ಲೋನಾಗ ನಮ್ಗೇನ್ ಬರಲಿಲ್ಲ ಅವರ ತಡ್ಕೋಬೇಕು ನಮ್ಗೇನ್ ಬರ ಅವರು ತಡ್ಕೋಬೇಕು.
Welcome back. I heartily welcome you all for the second session. So, just we discussed some of the basic research terminologies, and before that, the contents of how to write an synopsis. Next, the third point is synopsis writing in particular. So, I'll take some disease example, and for each heading, what is to be elaborately and pinpoint discussed under each heading, I'm giving clue by taking one disease in a synopsis writing pattern. Okay, so next slide. Synopsis. Okay. So here I'm covering 2 points at this juncture. One is synopsis writing. The second points in your syllabus research methodology. In practical aspect, there is one syllabus point. Synopsis bar dissertation protocol writing. Are you familiar with your syllabus points? Sure. Yes. You check it in practical. There is one point. Synopsis or dissertation protocol writing. That syllabus also here it is covering. The second point, how to write an synopsis, that point is also covering. Double action, double sim, like that. Okay. Next. So here, formulating the research problem, extensive literature survey, developing the working hypothesis, preparing the research design, determining the sample size, collecting the data. Next. Execution of the research work, analysis of the data, hypothesis testing, interpretation, then presenting the results, conclusion, and the publication. So at the end of your research completion, you have to publish that article. Let everybody knows what you did and what is the outcome. Let worldwide practice the successive remedies by the other doctors also. So the first point here, the formulating the research problem. Now you are at a stage. So before planning your research protocol, you have to select the problem. Either you are a Kaya Chikitsa or a Panchakarma or Shalya Tantra PG scholar. In which area you would like to do your research protocol? A Kaya Chikitsa can take a Sandhikatavata and other disease. A Panchakarma scholar can take Pakshagata, Ardhitavata, Gridarshi. Shalya Tantra person, they can take a different runas, different types of arshas and some ksharas, even some raktamokshana procedures, either a therapeutic and standardization also, because as Shalya Tantra is also concerned with raktamokshana, so even Panchakarmas also plan for the standardization of the procedure or the standardization of the equipments, modifications of instruments. So we have Shringa, Alabu, Jalauka, Gati Yantra. So in future days, we may not get this. So what modifications we can practice and expect the similar therapeutic effects over the patient. So you can think of and bring out new modified instrumentations. So based on your specialty, you can select a problem that is very important. Formulating or selecting the research problem is very much important. So what you should keep in mind, the researcher must decide the area of the interest and subject matter problem he or she wants to study. Feasibility and amenability of the particular problem or the solution before selecting the problem is very important. You should select the patient or the condition where on and offer the number of patients will report into this area or your hospital. That is very important. The second thing is your research protocol or the research problem. What are the investigations? What medications you are planning? What are the expenditure of that medications? Either it may be in a Grita form, Taila form, Shamanoshadi form, Rasayana form. How much duration? The feasibility of the availability of the drugs, its cost effective, its preparation, everything you should look into account. A patient, those are not reporting here and you are selected 40 sample size in a stipulated time, 1.5 years, it is very difficult to give justifications. That's why before selecting the problem, you should keep it in mind and plan or select the research problem. That is very important. Understand the problem thoroughly, rephrasing the same into meaningful terms from analytical point of view. So when you are selecting, you should understand and reframing the information. The best way of understanding the problem is to discuss your own colleagues or experts, senior researcher from government organization, administration agency. So before selecting, you are approaching your guide and the departmental teacher, sir, in which area I should work or what are the different areas I should select. So I have enlisted in the previous discussion the areas. So that is also very important. So the teachers or the guides, they have a lot of extensive experiences. They have seen a number of diseases. They have tried on a minimum sample size and they have their own observations and experience. So if you discuss with them, they will give some tips to opt different area, selective area where you can expect very good results and availability of the patients are very meet out the sample size is very much important. So all these should be kept in mind, and you have to approach this. Suppose if you want to do on corona post COVID also, this is one of the area where the different authorities, they have a statistical data. So you have to approach the government organization where with this geographical area or the Ballari district or headquarter, what are the population afflicted. So based on that, that will guide you to select a particular problem and plan. So these are all the things where you give the tips to opt your research problem. Example. So I am taken one example with Amla Pitta. Here I have not generally put Amla Pitta. So if you are suppose if you have written synopsis and sent just Amla Pitta, a question is arised in the remarks. Specify whether you are selecting Urdhvaga Amla Pitta or Adhoga Amla Pitta. That's why you should be prefix in particular. Whatever may be your problem, you should be specific. That is very important. Next. So that is one, selecting the problem. So after selection of your problem, what you have to do? You have to go for literature collection. Suppose if you have selected Amavata, so related to Amavata from Vedic literature to the Samhita Grantha and 21st century author, related to all the aspects of Ayurveda pertaining to Amavata, you have to collect, and contemporary related to rheumatoid arthritis and upgrades you have to select. And after collections, you have to extract one set of treatment. Either it may be Shodhana, Shamana, Rasayana. Which set of treatment you have to select, you have to extract. And you have to, then you finished two entity. That is one, you selected the problem, then you did extensive survey, collection of literature, then you are opting one set of treatment to advocate your treatment on a selected problem. The next point comes formulating the hypothesis. Yes or no? Yes or no? Following? Okay. Once problem is formulated, researchers should undertake extensive literature survey from all the context, academic journals, conference proceedings, government reports, books, earlier similar studies, library, internet, and article published in the index journal. So these will help you or support you to make relevant references for your research proposal. Next. So I will take an example of review of literature. Second point is review of literature. So what you have to highlight while explaining the qualities of Dugdha, Amla Pitta I am highlighting. Even the disease is not elaborately explained in Charaka Samhita, but we get the clues pertaining to Amla Pitta in a different context. The word Amla Pitta we come across. In the literature, we have to review. So Dugdha quality as explaining, it is Amla Pitta helpful in Amla Pitta or Dugdha. Amla Pitta Harana Shreshtaha. And Rajamasa is Patya in Amla Pitta. While describing the combination of Gora Anna Visha with Pitta, Amla Pitta is mentioned as one of the symptoms along with Daha, Krishna, and other Pittaja disorder. So these are all the literatures available in Charaka Samhita. I have given superscript 12. And at the end, what is the reference in detail I am quoting? That is called superscript. So you have to use in a different context where you are describing in your synopsis. So in Shushruta, these are all the informations available pertaining to Amla Pitta. I am highlighting and giving superscript numbers. So it should be serial one. So after writing 12, once again 5, 6, no. So you should go on increasing manner. So what reference you are selecting from a different text, go on giving superscript answer at the end. It should be written in the Vancouver style. The third point is developing the working hypothesis. For example, you have selected as a problem as Amavata, and you have selected, you did the literature, both the disease aspect and treatment aspects also, and you have selected Amrutadi Gugulu and Simmanada Gugulu. As you finalized that you are doing this Shamanaushadhi on Amavata, so you have to formulate the hypothesis to evaluate the efficacy of Simmanada Gugulu and Amruta Gugulu in Amavata. So then the third point is formulating the hypothesis. So H0, what you will write? H0, what you will write? Amruta Gugulu or Simmanada Gugulu are not effective in Amavata. Second one, Amrita Gugulu and Simanada Gugulu are effective in Amavata. This is one group. I'll change the similar treatment. I'll take Simanada Gugulu as a standard group and Amrita Gugulu as a test group. Then what may be the hypothesis? Null hypothesis, 0. Already one proved, Simanada Gugulu is proved. So I'm evaluating Amrita to evaluate the Amrita Gugulu is not effective over Simanada Gugulu because one is standard group. We are not evaluating because it is already proved and we kept as a standard group. That's why you should use the word over. Other you effects in North. You are not evaluating Simanada Gugulus because it is already proved and you have taken as a standard group. Followed? That is developing the working hypothesis. Next. So how does one go about developing the working synthesis? Just I told, discussion with the parents exposed about the problem, its solution, objectives in seeking solution, examination of data and record, review of similar studies on similar problem, personal investigations, original field of interview. So you will select the problem, you will go for some Shamana Aushadhi, you will take some Shodhana or Panchakama, and you will discuss with guides and your expert, you will approach your departmental teachers, take the relevance guidance. So this is the problem I have selected, sir. These are all the different remedies or the Shamana Aushadhis for the solution for the problem and how I should frame the working hypothesis. Then they will guide. So these are all the things already carried, and these are all the medications. So it is not mandatory that always one standard group is there. Even 2 new medications are also taken. So then the study becomes as a comparative clinical study. And 2 new drugs or Shamana studies can be taken and it becomes comparative clinical study. If it is one standard group and one new drug, it is named as under the title as a controlled clinical study because one standard group is there. And when you are advocating a new disease and a new drug, we can make it as a single group and we call it as a observation clinical study. But Rajiv Gandhi University very rarely giving importance for observational study. So basically, you plan for some control clinical study or comparative study. That is very important. If it is very new entity, new approach, and as far as possible, no studies have been carried out with the guidance of your guide. You can plan. You can plan a rare entity with observational study because. If you want to, even with the minimum sample size also, because no studies have been carried out and the patient availabilities are very less and your study durations will be very minimum. You cannot meet out in a short time with a large large sample size. So very rarely, if it is justifiable, if it is contributing to the society, new entity or new research field, you can make it as a observational study, minimizing with 20 sample size with the coordination with our departmental teachers, that can be framed. But better control comparative clinical studies are more authenticated for Rajiv University. Next. Scope study, why this study has been selected. So this I told problem statement. So here I've taken 2 medications in Urduanapita, that is Gudujadiquata and Rakshadiquata. My problem statement is Gudujadiquata is effective in Amlapitta, Urdunamlapitta, is Rakshadiquata is effective in Amlapitta, or is Rakshadi and Gudujadi. Rakshadi and Gudujadi are effective in to compare. Third questions or even third main aims and objectives it becomes. So it depends what medications and which disease you are taking. So why I am reading here, you have to give 2 lines because what are the contents of Guduchi? What is the Rasapanchakas, Rasaguna Vira Vipaka and its karma. So how it will play a role in Amla Pitta, that you have to give justifications in 2 lines. So that becomes problem statement. So based on this, you are framing the research question. So this is pertaining to Buddhachari and Drakshadi in Amrita. Suppose if you have taken some problem with some other medications, like this, you have to code with the problem statement and frame the research question. Next. Formulating the hypothesis. So, null hypothesis. Buddhachari and Drakshadi are not effective in Amrita. At hypothesis, Buddhachari is effective in Amrita. Drakshadi is effective in Amrita. Even you can extend one more hypothesis. This is more effective than this. That is more effective than this. So you can make 1, 2, hypothesis 2. So based on your medications, you can select it. The same thing, even if it is one Drakshadi is proved, we can consider as a standard group, and Buddhachari, we can take it as a test group. There are multiple options. So before planning, you should think whether I should take new 2 drugs, whether I should take one standard or one test group. So based on that, that will be helpful to frame research questions as well as hypotheses. So if you have taken one standard group, you are evaluating a new drug over standard one. Follow? Okay, yes. Okay, thank you. So preparing the research design. So in the introduction, as I told, single blind, double blind, triple blind, open, random. So which design would you like to plan? So that you have to put here. As I told, single blind, comparative, control, observational. So which type of step first before approaching this, you have to think. You have to decide. If you decide it, then it is very easier to frame this. So research design open, single blind, double blind, triple blind, single group, observational, double, comparative, and next control industry. As I told, one standard group and one test group. Even you can opt. Before to this, you have to think. So what my study decision should be, whether I should take one standard group or whether I should go for 2 new one. If it is a new entity, new area, you can go for some observational study also. Materials and methods, just you recollected in materials methods what I told. There are 3 entities: source, that is subject source, literary source, drug source. So the same thing. Pinpoint aims and objectives concerned to study effect of Urudaha and Urudaha. They are taken the Amlapitta, Urudaha, Amlapitta, the primary objective parameter to see the efficacy on particularly on the primary effect on the medication over Urudaha and Urudaha. To evaluate the effect of Buddhachari in Amlapitta, to evaluate the effect of Drakshadi in Amlapitta, to evaluate the comparative effect. Whether Buddhachari is more effective or Drakshadi is more effective. So don't go for aims and objectives to study the literature pertaining to Amrita. So n number of clinical studies researchers have been carried out. Then need not go for if it is new. For example, I will tell one hypothyroidism. So as such, there is no clarity in Ayurveda what is to be considered for hypothyroidism. Suppose if you are taking hypothyroidism, you can add because it is a contribution, it is a need of our. Because I am searching since 1 month. Hypothyroidism, modern and hypothyroidism in Ayurvedic parlance, you don't get information. So that is our contribution. If you do literary research on the hypothyroidism in Ayurveda, either it may be a direct one entity or we come across different terminologies. Galashoda, Pitta, Kahavruta, Udanavata, so like that. So you collect various informations which will mimic hypothyroidism based on its clinical parameters. You can consider. And even certain medications you look into, come across it is indicated in Ganda, it is indicated in Gandamala. And most of the drugs, they are processed with Ushna property, which will act as a kapha lekana, which is need of the hour in treating kapha. Or the subjects will be selecting of Amla Pitta will be selected and made into two groups, 20 patients in each group. Group A, 20 patients receives Gudichadi Kwata. Group B, 20 patients will receive Lakshadi Kwata. Next. So inclusion criteria. So on what basis you are including for your clinical trial, that you have to write. So you have to take some of the cardinal features present with Amla Pitta, symptoms of Amla Pitta like Amlodhara, Urudara, Taradara, Krishna, Brahma will be selected for the inclusion criteria. Patients of age group between 20 to 60 years of either sex or both sex will be selected. If you are taking any the Karma Shodhana treatment modality, the treatment is fit for your treatment modality. If you are taking some parasurgical procedures for concerned scholars, who is fit for that or parasurgical procedure, who is fit for that. Very important in the inclusion criteria. So patients exclusion, whom you are going to excluding patients having another Avashula and having major surgical illnesses, or those who are suffering with other systemic illnesses like diabetes mellitus, hypertension, and some other diseases, already taking modern medications which will interfere with your research medications. So all you are excluding for that. So once who is fit for Panchakarma or parasurgical procedure, need to write who is unfit. It is understood. And you have taken age 20 to 60. Need to write below 19, above 61. It is understood. Don't repeat. Don't try to avoid that. Criteria for a diagnosis. So for diagnosis purpose, what you are taking. So as the Amrita is concerned, there is no specific diagnostic techniques or investigation tools to pinpoint a diagnosis. Suppose if I have taken some Gridasi or some other like Ashmari, I will take the clinical features along with USG of pelvic or QB of X-ray, where we come across the presence of Ashmari. So that if you are selecting Ashmari in inclusion criteria, you have to write what is the size of the Ashmari, because you are doing conservative management. We have limitations. It's one of the acute abdomen where above 8 or above 10 also you cannot manage with Shamanaushadhi, because once if a patient had the Ayurvedic medications is very difficult to manage this. Then we have to take the modern doctor or painkillers you have to manage. So that's why you should restrict the size of the calculi, the number of the calculi, the place of the calculi, and you have to diagnose based on the certain investigations along with the signs and symptoms. Then that becomes subjective, the diagnostic criteria along with signs and symptoms, and the findings of EHG with the 8 mm, either it may be a single or a multiple, and this. So based on that, you will diagnose your condition. Withdrawal criteria. This is also very important. In earlier case, synopsis also we don't come across the withdrawal things. And when we are doing collaborative synopsis presentations in front of IC and modern doctor also, they are insisting, suggesting, and expecting that you have to write. If something goes wrong with the patient, either it may be with your treatment or the patient when they are coming to take your treatment, if something goes wrong, bad or unluckily, what you are doing, that the question raises. So in such conditions, if a patient is not able to take your treatment, you are withdrawing, and whatever the problems appears with your treatment, sometimes or with the other bad luck events, if something goes wrong, you are withdrawing and you are taking necessary care to the patient. Either it is possible from your side, and you are otherwise you are sending patient to the further centers to recover the problems. So that should be under the heading of withdraw the criteria. That is very much necessary to write down. Dosage, this is very important. The good chai water, 20 ml twice a day. Here, 20 ml is the dose, equal quantity of water, that is anupana. After the sevana before food, anupana water, duration 30 days. It should be specifically written. These are all very important and clarity. Next is Dakshadi water, 20 ml twice daily. So always your group should be identical. Your dosage should be identical. Your study duration will be identical. As per my knowledge, Katibasti is not comparatively aggressive with Abhyantara Basti. Do you agree or not? So always your treatment should be identical. Don't compare one Abhyantara Shodhana Panchakarma with Baya Basti. My knowledge was it should be always uniformity or identical, unbiased. Unbiased. So Baya Chikitsa should not be compared with other treatments, but you can compare two different formulations of Bhakti Chikitsa in single condition. Like Vaitarana Basti and one more Yerandamula Basti in Grudarsi Vasta. To evaluate the efficacy of these two Bastis in Grudarsi Vasta. But you cannot compare Kativasti and Yerandamula Basti efficacy because they are not identical group. One is Abhyantara and one is Bahya. So that's why your treatment protocol should be identical, dose identical, duration identical. Follow study duration should be identical. Then it becomes chaos this study. Parity. Okay. Next, total study duration will be 60 days. So in 60 days, on which day you are going to evaluating your efficacy? 0th day, 15th day, 45th day, and 60th day. So if your assessment will be on 0th day and 60th day, we have to take the readings. We have to take the readings because in which you cannot take because your treatment duration will not be completed yet, and you cannot expect the results. And some treatment, particularly in Panchakarma, after finishing your Shodhana the next day, you cannot take the evaluation or assessment of your results because the Shodhana treatment modality is having its long-lasting effect. So you should finalize in the follow-up period because it is slowly relieving its efficacy and the patient will get still better improvements in a follow-up period. So that on which days you are assessing your result, you have to. Even on the 15th day, you are not taking the assessment, but what may be the improvements, what may be the merits, what may be the demerits you should observe. If something goes wrong, what you did for each patient, that patient observations you have to write. This is the reality of your research work. You are following what I am telling. Patient number 1, the end one page. On 15th day of medicine, either Shodhana or Shamnaushadhi, what are the good things, what are the bad things? Write down the demerits. If patient something went wrong, what you did. With Shamnaushadhi, the patient had vomiting sensation or burning sensation in the poster. For this, what you did, that you have to write down. This is the outcome of your research work. This is called observation on various steps of your clinical study. Observation on various steps of your clinical study. You have to observe the patient doing amalgam himself. If you have given trigger to churna, the patient had gastric erosion. What you did then? You have to write down observation. Will you withdraw the patient? What will you do? What will you do? So you have to minimize the dose. You can increase the quantum of anupan and terms of the patient. Then the adverse effect or the whatever the minimal complications or adverse we observe that will be get subsided. Then post Ashokana, what patient had taken, how many has he has once, twice, thrice, or 20 times. For that, what you did, 20 times also patient is better, or the patient need IV lines. Whether you started IV lines or what you did. These are all the reality of outcomes. But what is the fate? The scholar will be in his or her native place. The guide is receiving the patient. The guide is giving the examining the patient. The guide is giving the medicine. Some guides are exhausted and research scholar in their native place. Patient medicine next scholar next ಇನ್ನೊಂದು ಸ್ಕಾಲರ್ ಹೇಳ್ತಾರ ಏನಾಗಿತ್ತು ಮಳಿ ಬಾಳ ಆಗತಿ ಅಲ್ಲಿ ಬ್ಯೂರೋದಾಗ ಮೆಡಿಸಿನ್ ಇದೀನಿ ನೀ ಕೊಟ್ಟು ರೀಡಿಂಗ್ಸ್ ತಗೋ ಸ್ಕಾಲರ್ ಊರಿಂದ ಬಂದು ಆ ಕೇಸ್ ಮುಗಿದು ಹೋಗುತ್ತೆ. This is in reality the research processes are going on. This is not ethical research. This doesn't have any outcome. So when you need, then day one and after completion, for each step of your clinical study, you have to observe the patient. Sorry. Am I right or wrong? It's huh? The patient is somewhere, the research scholar is somewhere, and is struggling to complete the total sample size. Huh? ಯಾಕಂದ್ರೆ ನೀವು ರೊಕ್ಕ ಕೊಡ್ತೀರಿ. ನಾನು ಗೌರ್ಮೆಂಟ್ ಕಾಲೇಜಲ್ಲಿ ಕಲ್ತೀನಿ, ನಾನು ನಿಮ್ದಂತ ಬಟ್ಟಾರೆ ಹೇಳಕತ್ತಿಲ್ಲ. Generally speaking, generally more of the research institutes this is going on. ಇದನ್ನ ಆಗೋದಿಲ್ಲ, ಡೆಸರ್ಟೇಷನ್ ತಂದಿರ್ತಾರೆ, ಇನ್ನೂ ಸ್ಟೆಪ್ ಮುಂದೆ ಹೋಗಿರ್ತಾರೆ. Why I am telling means the research should be genuine. There is something contribution to the Ayurvedic fraternity. You should feel pleasure. You should feel pleasure that I did work on this and I have some ideology, some contribution from my side to the Ayurvedic fraternity. Yes sir. Ayala ಮಾಡ್ತೀರಾ? Start. Day 1, 2 until last day of follow-up, the patient should be in your supervision. Then you note down the outcome of your first patient's positive, negative, or merits and demerits, and that should be added to the discussion part. That is real contribution. You should feel pleasure, not pressure cooker, with your study. So this is possible when you dedicate it total to your research work. Like what I am on to highlight. Next. So here, these are all the content parameters group A and group B. Just I kept emptiness based on your research protocol or the proposal selected. You write down samples, Amapachana, coastal Shodana, procedure and medicine, type of basti, dosage, Anupana, duration, Aushadha Sevana Kala, follow, study duration. Based on your branch specialty, your protocol proposal, you write down. You should not omit any of the heading. It should be clear. Otherwise, in synopsis evaluation, the dose is not clear. Aushadha Sevana Kala is not mentioned. Anupana is not mentioned. Follow period is not mentioned. Basti, so these are all the remarks attend and incorporate during your trial. These are all the remarks. But if you clarify now itself, there is absolutely no questions, only good remarks. What good remarks? Your synopsis is provisionally registered. Happy. Provisionally registered. Remarks? No remarks. Otherwise, attend 6.1, attend 7.2. Your encore still are not in encore. These are all in number of, and you will get attention. So if you attend all these things, 100% your synopsis will be provisionally approved. Assessment criteria, just I told, you should have certain objective parameter. Security. Work no, sir. You should put your efforts day and night. Other than the lot of objective parameters. That is very important. So, you select such studies which carries more weightage. You can show before treatment and after treatment with certain evidences. Then you will have certain confidence, and your study will get a lot of recognition. So that is very important. Based on your sample size and group, you have to be specific. Whether you are applying t-test, paired or unpaired t-test, ANOVA test, blood test. Statistically analyze with relevant statistical data you write. No, earlier 5 years, 10 years, that will be possible. But now, remarks will be which statistical test you are applying, be specific with the remarks. So if you are selecting 2 groups, sample size, paired and unpaired t-test will be applied. If you write down, that question will be solved. Investigations, this is very important. Now you are selecting research protocol. Try to be specific diagnostic investigations rather than in general. It's specific diagnostic. Even sometimes screening also we used to go for CBC and some RBS also. So these are all certain conditions. If you are selecting Mutrashmari, USG pelvis, BT80, Shrinachukra, semen analysis, BT80, Madhumeha, the latest HbA1c, based on that, if you select that, there is more weightage for your studies. Next. I have given some examples. You are the new research designs. So observations during Apachana, during post Shodhana, Vamana, Virechana, bus, any other parasurgical procedures, Shama Chikisa, Rasayana, Patya, anything what you have, then follow it. So you have to write down observations on various steps, and that should be utilized for the discussion of total 40 samples. This is the reality and outcome of your research. Next. Oral assessment. Next. So determining the sample size, just I told how you are deciding the sample size in 1.5 years. So complete your sample size. What will be the total sample size you should decide? 100 sample size if you want to take and finish it in 1.5 years, it is very different. Or usually 100 sample size is mandatory. 90 is not sufficient. Beyond that, you can take. Why? It is extensive study. The duration will be little more, 3.5 years. Total 5 years. It is partial fulfillment or doing it other institute. 5 years will be there in first year examination. No need to go for this. And when they initiate in the second year, they should start with clinical trial. Minimum 3.5 years they will have time. That's why the sample size is more. So in Ayurveda, why the sample size should be more is if the sample size is more, the accurate the results. So if you do the larger sample size, you can state the statement exactly. So that's why with the smaller sample size, you cannot conclude. So that's why in PhD, larger sample size is mandatory, minimum 100, and beyond that also you can take. So how many sample size? That is, I told what is the problem you have taken, what are the total number of patients reporting to your hospital in annually, and in 1.5 can you meet out that sample size? So all should be taken into account and decide your sample size. You will read different sampling techniques. There are simple sampling techniques, ready-made cluster, quota, multi-stage, multi-phase, all in research methodology. You will come across. You will familiar with these. Next. So collection of the data. So when you are collecting your data, so you started your treatment day one. A patient comes to you, based on your problem you have selected. You will have the gradations. It's the severity. In the day one, you will take the data. And suppose if your treatment will be for 30 days duration and 15 days follow, 0th day and 30th day, you will take the data. And even if you added before treatment, after treatment, and after follow-up also, you will take the details or the data of after follow-up. So the data should be collected and it should be calculated when you want to go for statistical analysis. And through personal interview, through telephone interview, questionnaire, through schedules. So there are different methods. You will read it in a research methodology. Through interview method, some certain questionnaire also will take the data. So based on your study, what you are, if you have certain standard, certain scales, there will be a questionnaire. So when a patient comes to us, if the patient is educated, we'll give the questionnaire list. Otherwise, if patient is uneducated, we'll ask the questions and tick as the relevant criteria. These are all the different methods where you are analyzing the data. So execution of the results. So execution of the project is very important in the research process. The execution of the project proceeds on correct lines. The data to be collected will be adapted and dependable. The research should execute in a systematic manner. So how you want, you cannot take the data or execute it. So we submitted our research protocol, research proposal. So in a systematic way, when we are initiating the treatment and when we are analyzing this data, soon after completion of our treatment, then 1 month follow-up is there. Directly, you should not tell come on 30 days. The 30 days also divided into 2 parts, 15 days once. Even if it is a 15 days, weekly twice, week 2 weeks means 1 week. First follow-up, second follow-up. So to keep touch with the researcher and the patient, we should weekly or 15 days we should have follow-up. Otherwise, the patient will not come to you because patient don't have time. Yes or no? Where is what happens? The doctors are adjusted with the patients. Patients are not adjusting with this. Sir, Panchkarma, basti mane ಕೊಟ್ರಿ ಸರ್ ತಗೋತೀನಿ ಹಿಂಗಾಗಿ ಬರ್ತಾರೆ ಮಾಡ್ತೀರಿ ಎಲ್ಲ ಫ್ರೀ ಕೊಡ್ತೀರಿ ಆಯಿಲ್ ಟ್ರೀಟ್ಮೆಂಟ್ ಅಲ್ಲೇ ಇನ್ನೊಂದ್ ಕಡೆ ಇಲ್ಲ ಡ್ಯೂಟಿ ಹೋಗ್ಬೇಕ್ರಿ ಮನೆ ಮುಂದೆ ಯಾರಿಲ್ಲ ಮಕ್ಕಳಿಗೆಲ್ಲ ಬುದ್ಧಿ ಮಾಡಿ ಕಳಿಸ್ಬೇಕು ಕೊಟ್ಬಿಡ್ರಿ ನೋಡ ಕೊಡ್ತಾರ ಕಾಮ ತೋಟ ಪಂಚಕರ್ಮ ಇದು ಒಮ್ಮನ ಮನೆ no, it is not possible. Even then we don't want to miss our patient. ತುಪ್ಪ ಸ್ನೇಹಪ ಮನೆಯಲ್ಲೇ ತೋಡಿ ಒಂದು ಪೇಪರ್ ಅಲ್ಲಿ ಎಲ್ಲ ಬರೀತೀವಿ ಬ್ಲಡ್ ಕ್ಲಿಯರ್ ಮಾಡಿ 30 ml ಚೆನ್ನಾಗಿ ಅಷ್ಟು ಗಂಜಿ ಗಿಚಡಿ ತೋಡಿ ನೆಕ್ಸ್ಟ್ 50 ml ತಗೋರಿ ಅಂತೇಳಿ ಎಲ್ಲೋ ಸ್ವಲ್ಪ ಗೈಡ್ಲೈನ್ಸ್ ಮಿಸ್ ಆಗ್ತೈತಿ. ಮನ್ಯಾಗ 100 ml ತಗೋತಾನ 11 ಗಂಟೆಗೆ ಒಂದು ಫೋನ್ ಮಾಡ್ತಾನೆ ಸರ್ ನೀವು ಅನ್ಕೊಂಡ್ರಿ 10 11 ಕ್ವಾಂತಿ ಆಗ್ಬಿಡುತ್ತೆ. realities. When we will achieve success, when a patient under the supervision of the physician. ಏನಾಗ್ತಾ ಇದೆ ಅಂದ್ರೆ ನಾವು ಪೇಷಂಟ್ಸ್ಗೆ ಅನ್ಕೋತಾ ಇದ್ದೀವಿ, ಪೇಷಂಟ್ಸ್ ನಮ್ಗೆ ಕೇಳ್ತಾ ಇಲ್ಲ. ಪಥ್ಯ ಸರ್ ಊಟ ಆದ್ಮೇಲೆ ಒಂದು ಊಟ ಕಂಪ್ಲೀಟ್ ಆಗಿದೆ, ನಾವೇ ಬಾಳೆಹಣ್ಣು ತಿನ್ಬೇಡ ಅಂತ ಸ್ನೇಹ ಕಾಲದಲ್ಲಿ ಸರ್ ಯಾಕೋ ಬಾಳ ಇಷ್ಟ ಅನ್ಸುತ್ತೆ, ಸೈಡ್ ಒಂದು ಉಪ್ಪಿನಕಾಯಿ ತಗೊಳ್ಳಿ ಊಟದಾಗ ಸ್ವಲ್ಪ ಮೊಸರನ್ನ ಉಂಟ ಅವತ್ತಿಂದ ಊಟ. ನಮ್ದು BP 120/80 ಇರ್ತೈತಿ, ಉಪ್ಪಿನಕಾಯಿ 130 ಆಗೈತಿ, ಮೊಸರನ್ನ 150 ಆಗೈತಿ, ಬಾಳೆಹಣ್ಣಿಗೆ 160 ಆಗೈತಿ. ಯಾಕೆ? The patient is under supervision. The patient is not following pathya. Rather than getting improvement, the complications are more. ಅದಕ್ಕಿಂತ ನಮ್ ಟ್ರೀಟ್ಮೆಂಟ್ ಇಲ್ಲ. ಅವನಿಗೆ ಗಮನ ವಾಂತಿ ಆಯ್ತು ಸ್ನೇಹ ಕಾಲದಲ್ಲಿ ಅಂದ್ರೆ ಮತ್ತೆ the total schedule will be interrupted. So that's why we should take care of this. Whatever treatment you are giving, convince the patient what treatment you are giving. Take into the concept. This will be help for a desired effect, very good results. I mean, patients were doctor, the patients will be very happy that you are very closely related to the patient and you are interacting with the patient. Then he will send a few patients. Hey, very good physician is here. Come with me. I will introduce. So how you are giving approach to the patient, and you will have a good names. So these are all very much necessary for a new researcher or physician for their future also. So analysis data, as I told, before treatment, after completion of treatment, and after follow. Severity, grade general normal, severity is 1, 2, 3. So this will be generally we give the data, and after treatment, what will be the severity? If it is 4, after treatment, what will be the gradation, and after follow, what will be the gradation? Patient ಬರ್ಲಿಲ್ಲ 4 ಇದ್ದು after treatment ಮಾಡೋದು, after follow 1 ಮಾಡೋದು. So ಇದೆಲ್ಲ ಮಾಡಬಾರದು. So you should analyze. You should analyze the patient. 4 gradation, if it is as severity will be the same. Give justification. What may be the reason? The severity of 4 gradation will be comes down in other patient. Why in this patient? Make it a note. Patient is not taking the proper medications in time. Patient is not followed appropriate pathya. Patient is not came out of the follow. Patient is not Biostatistics. These are all the things where we cannot expect. Maybe the patient is not improved on this parameter. What you are observing, you write as it is. In reality, don't go for fabrications. Next. Hypothesis testing. So you framed H1, H0, H1, H2. So based on statistical analysis, you will get p-values. Whether p-value is significant or non-significant. If it is not significant, your hypothesis will be H0. That Drakshadi and Gudchadi are not effective in Udvara Amla Pitta. If p-value is significant, your Drakshadi and Gudchadi are effective. Following what I am telling, hence these are effective. My research hypothesis will be acceptable because p-value is like this. Followed? So interpretation. Even if it is good outcome, even if it is not improvements, you have code. As I told, what may be the relevance? Certain parameters we found very good results. In certain parameters, there will not be much improvements. What may be the reason? You give your interpretations. In various tests of your clinical study, that is the interpretations of your research. This is very important. Preparation of the report of the dissertation. So you started your clinical trial, synopsis, dissertation, pattern. So you have to write down. Means in the last 6 months of your examination, you have to prepare your dissertation. So there is a certain protocol to write a dissertation. I think you'll come to know in a second. So standard guidelines will be there. how to write in a desert. So they have specified already. You did all this. You have all the data. You should be prepared with the dissertation protocol. Introduction, abstract, introduction, metrics and maths, like this review of literature, drug review, like that. There will be certain the contents to write the dissertations. So whatever you did, there is a certain standard protocol. You have to frame the whole research information under the heading of dissertation writing. Prepare the report what you have done. I think the reports must be done with a great keeping with the view. So what we are doing, we did all these things and keeping our records in our locker. No, this is not the end. So it should be proceeded further. So you have to extract whatever the contribution you did. You prepare around a 10 page. One part of minute test information about iteration and materials and methods. So what you did, what medications you have done, what are the sample size. There is a process of scientific research article. So under your guide, you have to save and with publications, you have to send with the pre-reviewed recognized journal. So let your study research information will circulate worldwide. It is really contribution to the society. These should be practiced by everyone. We have a journal, we call it a journal review. So we used to take this published journal and we will discuss in the PGT part. So this is also one of the contribution from your side. Your article will be read in the journal discussion, means that is also one of the contributions. There also we discuss merits and demerits. No, some errors will be there that could be upgraded, but it is very good. Merits and demerits will be there. But if your article will be read in the one recognized institute in a journal review, that is also one of the contributions. And let other take as this your article will be the one reference for their forthcoming research. Followed. So, paragraph writing this dissertation, abstract, introduction, summary of the findings based on what you observed, main report, conclusion. So, these are all the structure how you should write the dissertation. So, there will be standard guidelines from Rajiv University. When you enter to the stage, you will get that from the department. So, try to avoid what I feel, maybe, probably, I think, my opinion, from my Ayurveda, my Acharya, my teacher, I seems, all these such words should be avoided. Be clear. Don't use the formulas like water means H2O, increasing arrow mark up, decreasing arrow mark down. No, such abbreviations, such formula. Be clear, simple. Don't use complex, very ready to understand. Don't use simple. Have a new kind of English subject don't go for standard, simple. Ayurveda, simple, clarity. It should be free from ambiguity. We use the word ambiguity. Ambiguity means a lot of confusion, no clarity. Your terms should not be confusing. What I feel, my friend told, I think. So try to avoid. Clear. Next. So to sum this protocol, formulating the research problem, extensive literature survey, developing the working hypothesis, preparing the research design, determining the sample size, collecting the data, execution of the project, analysis of data, hypothesis testing, interpretations, preparation, presentations with the results, conclusion, and application of your. So this is the total reviewing or summarizing how to write a synopsis. I use some titles and protocols. Now engage. I'll give some of the protocol how best we can frame. You may query. For ideology, I'm giving something formulated and some already did. You have to modify. Take this idea, formulate your own. Okay. Protocol 1, comparative clinical study to evaluate the efficacy of Aditya Pakagugulu and Yamagugulu in managed data with special reference to osteoporosis. Don't write W R. Keyboard. Everything should be in detail. Fully. No, no bargaining or no conjures. Right, full as a whole. So what are the two parameters if you are considering as a research product? You can take for bone mineral density, then serum calcium level as a objective parameter. Rest others will be there. Before treatment and after treatment, if you do this, you will have some good weightage for your study. Protocol 2, comparative clinical study to evaluate the effect of Madhusri Rasana and Chavaputika in detail special reference to psoriasis. So as psoriasis scale severity assessing index is there, it becomes one of the standard assessing scale without any hesitations. Before treatment or treatment, based on this, you can assess the efficacy of your treatment. Right? Protocol 3, comparative clinical study to evaluate the efficacy of we have been behind in the management of food as a Rasayana effect also you can select. Some Rasayana objective parameter, that is also one of the new contributions. Control clinical study to evaluate the effect of yoga Rasayana over Namayasaloha. Means Namayasaloha is already proved, but we are taking as a test as yoga as a Rasayana. And what are the objectives we are keeping? Hereditary anemia, serum correcting, or blood pictures. Any one of these you can take before treatment and evaluate your medications after treatment and you what is the outcome. A single blind randomized control clinical study to evaluate the effect of Patarigulu or PARYESHANA. This is one new protocol I framed. Here you come across three groups, one standard group and two test groups. If your protocol will be having three groups, no doubt sometimes majorly don't have only two groups, and there is nothing wrong if you take three groups also. So in that you can take one test group, one standard group and two test group. So you are evaluating two yogas over the standard group. So this is particularly as I told, primary outcome or objective parameter MRIT and AT. You are evaluating your medications. Even you can add or Basti followed by Shamana also, no problem. Next, even changes on SLR. What is the day one, the degree of pain, and after completion of your treatment, you can assess that also consider. This is one more slide. So, SLR or as a diagnostic X-ray, you are not taking after treatment also. But there is one Rona Morris disability scale. So this is one of the standard scale where you are assessing the severity of the brothers, particularly. So if you select this, it carries more weightage for your study. So I would like to suggest this slide. Visual analog scale. So this is one of the standard scale to assess the severity of the pain. So we have a standard guidelines where you want to assess, evaluate the pain. So you can incorporate this standard scale. So one more protocol I would like to highlight. Evaluate the effect of Panaspatra Sweda, Shiruma, Ekang Vilvas in Aditwaka with special reference to health policy. So here two groups are there, two different procedures are there, Shiruma and Panaspatra. Ekang Vilvas is common for both the groups. So this is one of the protocol and researcher name, grade, and your college name. So these are all needed for the study from the physical science. Superscript I have underlined. So what materials information you have taken given number and underlined, you have to write down the references taken from that. This is as I told problem statement. What your procedure or medications taking, what are the pancha rasas are available, gunakarmas. Try to refer Indian medicinal plants volume 1 to 5 by G.S. Levaker. Those who are taking any medicines in the form of formulation or Basti, in this text, from all the information of a single drug you will get, specifically the pharmacological action, chemical compositions, and active principles. Suppose if you want to go for Bala and Bala, if you want, all the information in this context you will get, including chemical composition, active principles, pharmacological actions. So in a single platform, you will get more information which you are not concentrating with other texts. So better if you concentrate, you will get all the information. Review of literature. Where are all the literature concerned to Aditwata, Kshiraduma, Panasapatra, Ekang, has been highlighted in Samhita Granthas. I have given. So, principles and practice of medicine. Don't write in explanation all textbook names. You should give superscript and the last page. Next. Previous work done. Particularly, in the study of the special of now is the it is also called Shira Bhash Sveda. Mine is Shira, mimicking, saying, communication. Clinical study to evaluate the effect of Shira in the management of Aditwata in this way. It is equal to Bhash study. So you have to take Balak Shira. Instead of Shira, study has been carried out Balak Shira. So you can take modify other, but you can take it. 2.8. This is very important. I'm insisting to write down. This is the specialty. Please make it known. Usually, I advise PhDs. If somebody asks why you have in IC, you should ask why you have selected this study. What is your contribution? What is your opinion? So I guide you. Note on this. For example, if you have selected one disease, two treatment modality. You browse different research portals. PubMed, Dara, Ayush portal, Scopus, even you can add some more. And justifying with Aditwata, Ekang, Panasapatra, Kshiraduma. In PubMed, how many related articles I may get? Dara, Ayush portal, Scopus, Ekang, how many studies has been carried out? Kshiraduma, how many researches has been carried out or publication article? Related to best policy, how many things? In majority, you come across zero, and Aditwata or best policy will have a lot of information pertaining to literature. Aditwata best policy we get 550 research information in PubMed. Absolutely no studies, no articles, no dissertations. Very, very minimum. This is create an interest to select this area and take it for the research work. You frame for your work, find out and frame for this. This will give a lot of information to the IC members and evaluator of synopsis also. Usually it will not be followed. So I used to suggest my scholar, my PhD scholar and undergraduate staff, PG scholars. But PG scholars, if you find there is nothing wrong, you are upgrading your synopsis. You are upgrading your synopsis and quality of synopsis. So I would like to highlight. Research question: all three medicines is they are effective in Ardita Mata. Hypothesis, null hypothesis, and alternative hypothesis. Single procedure with the Shamanaushadhi, other procedure with Shamanaushadhi, and comparing which one, group A, group B, comparative efficacy of either A or B, we are evaluating. Next, that is aims and objectives. To compare the effect of Ashwagandha and individual and comparing the effect. That is aims and objectives of research. This research program. Materials and methods, as I told, subject source, drug source, literary source. You are familiar now? Source. Preparation of the medicine, identification, preparations, where the yoga is mentioned and where you are preparing, you have to state the information. Study sample size and grouping, sampling technique, simple random sampling technique. Or if you are using any computerized technique, you can put. So don't go for general statements. Be specific. Inclusion criteria, signs and symptoms of with positive Bell's phenomena. That is very important for the diagnosis purpose. Next. Subject associated with Pakshagata, I am not taking. That is exclusion. This is chronicity of 1 year. Next. So diagnosis based on Bell's phenomena positive along with the signs and symptoms of Pakshagata. Group A, 40 patients, 2 groups, 20 patients in each group. Group A receives this set of treatment, Group B receives this set. And total study duration will be this much, follow including. Okay, so this is the pattern of writing postology. Next. So briefly about how you are preparing and how you are applying. In brief, not in detail. Okay, next. Even if you are not writing, but you should be able to answer the question asked. How you are performing the procedure and on what basis, what are the references. You should have the knowledge. Even if you are not written the synopsis, you should have an idea. Subjective parameter, objective parameter. So these are all based on previous work or previous dissertation based on sentence. That's all. But later I have added the scale assessment parameters. Brightness scale and facial disability index scale. So these are all standard parameters to evaluate the efficacy. So these are all the relation. We have to ask these questions and put the details, and you have to calculate BT, AT, and EM. So based on that, we can say our efficacy of our treatment. Next slide. So if you use or find out according to your protocol, it carries more weightage to your study. Continuation of different assessment plan, social information. So some objective. Usually what we do when we are a PG scholar, we try to remain dissertation similar and keep as it is. So sometimes the authority will not accept. So they want standard. American rheumatoid criteria, Brightman index, OMAC, VAS, ASCII, these are all be accepted by the authority. So if you tell sir previous scholar has taken, sometimes it will not be acceptable. But if you work still more, that can be taken as a secondary parameter. But Brightman scale and FDI to be taken as a primary objective parameter. This you can take it as a second. So these are all most common. There is no specific investigations to pinpoint diagnosis. I diagnose based on health phenomena along with signs and symptoms. There is no particular investigation. So if necessary cases, you can go for CT3. These are the list of references as per the important. The last page number, if you see, yes, it is right. Coastal written by these are the references. Some of the research protocol we can think of clinical study to evaluate the we are getting one Abhyantara and one Upanaha. So these are not acceptable because Sweta is not comparable with Abhyantara. But two different Basti Kalpas can be compared. So that's why it should be unique, identical. Treatment should be identical, posology should be identical. Evaluation of the efficacy of Potential and the Lavana Portale in the Sweda in the management of Amava. The two both Swedas are Ruksha type of Sweda. Yes or no? So that's why I'm giving clarity. Not generally Sweda, Potential Pinda Sweda and Lavana Pinda Sweda. That too, our agenda. That's why we are specifying. Right? Comparative study with Bunaga Thailanese and Karpasa Thailanese in Ardita. It is an identical group. The treatment will be on the same route, but different kalpas. We are comparing which Thailanese is effective in Ardita. Evaluation of efficacy of Sharavasti and Vaitana Basti in the management of Amavata. Comparing two different bastis in a single disease. A single blind randomized controlled clinical study to evaluate the efficacy of Gurchand Yoga, Pachand Yoga over PARYESHANA Yoga with Ruksha Vinechana in major Yoga with special reference to hyperlipidemia. Here, a new protocol. Here, you can expect 3 groups, and there will be one standard group, and we are comparing that group 2 new drugs over the standard one. If your trial group study protocol will be having 3 groups, so this you can take as one standard group and 2 test group, and you are evaluating 2-1 over the standard one. Single blind randomized controlled comparative clinical study to evaluate the effect of Tritiya Tikla Rasayana, Valamula Rasayana or Avadi Chona with Panchatika in the management of Astic structure process. For 3 groups, Asti is common. 2 3 different Shamanaushadis we are adding. A, B, C. Which one is more effective? Here, one standard group is there. 2 new groups are there. Test groups are there. You are evaluating 2 new groups for the test standard group. Single blind randomized comparative clinical study to evaluate the effect of Pathya and Vata in the management of the same thing. Simple two Shamanaushadis we have taken. Those are new and we are comparing. Or you can modify one standard group. If it's already proved in Kadrasi, you take one new Kalpa and it becomes control clinical study. Now it's time to have a discussion, a questionnaire. If you have any doubts, clarifications, you can ask without any hesitation. The question may be wrong, I'll make it correct and I'll try to solve your problem. Any questions? Without any hesitation, you can ask simple, complex questions. What you try to answer, what your facing hurdles or problems or not clarifying information. Questions no problem. It is really needed for our context. Why I'm telling because my and I need the customers and I am and because of stress and depression, since 20 years we have but I find out any upgraded versions like they from that you can take. So, record of the what are the upgrades and standard parameters you can consider for this house. There's nothing wrong in it. So I will tell you because I will all the activities so I used to do all there's nothing wrong in it. Just there is no room to expect others sometimes because they don't have basic ideology, they don't have our principles. We try to convince them. Acceptance is from their part. We feel satisfaction and we have confidence and we have the ability to learn results. First, we agree to your guide and teachers and your friends. Please accept. Don't go for internet. Normally, I will accept properly. You know, I will be this person. I will be medicine properly. Yes, they are coming over the level. Yes, they are Vishwasana Bhakti. Yes, they are Abhinam Bhakti. This is like now we enter. Now over Bhakti. Now I will now confidence for. There are not acid for you. Yaru. English programs for you. English for ashwagandha, anything. Commercial ಆಗಿ ಯಾರು ಕಷಾಯವನ್ನು ಮಾಡಿದ್ರು ಅಶ್ವಗಂಧವನ್ನು ಮಾಡಿದ್ರು ರುಚಿಯನ್ನು ಮಾಡಿದ್ರು 1964 ಅವರೆಲ್ಲ
yoga on Google as a total inactivity will not be acceptable. Like that, when we sit in front of the ethical committee, please. So, the yoga, which is consisting of different ingredients having its efficacy on the particular problem. One Biostatistics is effective in particularly pain.